The topic of this the international conference, they have selected the latest development of the dosage forms. That is the personalized medicine. So this is the latest trend of the dosage forms, and this topic
kits are available in the world and 300 types of drugs are available for the treatment of some personalized patients. A really wonderful project. And in course of time, what is the benefit of this? What is the use of this particular research? The discovery of the drug, which is useful for a particular patient, will be mentioned by doing this. Suppose if a patient is diabetic, can we prevent diabetes in this case? That can be done by genome project, genetic counseling, genetic therapy. That comes in this review. And also, by this, we can have some solutions in future. Treatment for cancer where there is no answer for this cancer, Alzheimer's disease, hypertension, some of the diseases where we cannot find any solution, probably by doing some research in future, we may find about these trends. We are now going to have discussion in the coming 2 days, and the people of eminence will be going to speak about this. And also, on this occasion, I suggest that graduates of B Pharmacy are not coming up to the level of international standards. Why? There is some lacuna in the teaching methods or the universities or the colleges or institutions where there is no modification or there is no intention to go for competition with the international level. And it is suggested recently about the graduates are to be competent to the level of international level. Graduates, just B Pharmacy students. If you go to M Pharmacy, okay, it's research oriented and all that. At B Pharmacy level, it is suggested that first 2 years of study should be of basics. Then you have to choose a specialization, aesthetics, pharmacognosy, just like that. That's what it is suggested. If it comes to the, I request our president also to think about this so that the graduates will be competent when they go into society, that they will be honored by the society. Once they are getting this type of knowledge, either the pharmacy practice, pharmacy, clinical pharmacy, or community pharmacy, all these things are specialized subjects which are to be learned in the later 2 years, that is in the 4 years of curriculum, the third and fourth years are to be taught. So, and also, I want to suggest about the ecosystem, that is about the inspire, innovate, and incubate. 3 I's. Inspire. The persons who have got vision about having a pharmaceutical company has to be invited, and they are to be invited to give some support to the institutions so that there will be some research that will be going on by the institute with the help of, with the support of some visionary, that is another company, pharmaceutical company, so that some product will be produced by that. That is incubation. When the products come out to the market, then by that there will be some gaining of some economy. This economy has to be again put into this practice, should become a cycle, so that there will be further development of these the pharmaceutical industries, pharmaceutical sciences, institutes, and knowledge about all these things. So there are the few things which are coming up. They are to be thought of, and I request our president of PSA to introduce all these things into our curriculum in future, so that the society of the institutions, the society of India also will come up with good standards. Thank you very much for giving me the opportunity. Thank you very much.
Thank you, sir, for your inspiration. It's a pleasure to invite Dr. SLN Prasad Reddy, sir, member, Central Council, PCI, to share few words to the gathering. Sir, please.
Andarki Namaskaram. It's indeed a pleasure to be here among the distinguished people here. First of all, I should convey my regards to the distinguished people on the dais. Dr. Montu Patel, Pharmacy Council of India President, and Dr. Lau Rattaya Garu, Chairman Vignan Group, Dr. M. Venkatramana Garu, Central Council, EC member, PCI, Dr. Niranjan Babu Garu, CC member, PCI, Dr. Radha Krishnamurthy Garu, CC member, PCI, Dr. William Carey Garu, the Registrar, AP State Pharmacy Council, and my teacher. Then I like to convey my good wishes to B. Srinivas Garu, the principal of this college, P. Saujanya, convener of this program. And my regards to all distinguished speakers sitting on the dais and off the dais, distinguished people of the dais. My first and foremost, on behalf of the pharmacy fraternity of AP State, I must welcome the young, energetic PCA President, Dr. Montu Patel ji, to our state, Andhra Pradesh state. As you all know, the crucial part of any nation healthcare system consists of mainly three parts. One is the diagnosis of the disease or disorder. Number two is the pharmaceutical treatment. Number three, that is complemented by the service of the nurses. But here, we as a pharmacist play a crucial role. We alone plays a crucial role in the pharmaceutical treatment, which is a crucial part of human healthcare system. The goal of any pharmaceutical individual, either he may be a manufacturing pharmacist or a retail pharmacist or a drug regulating authority like PCI, a drug controlling authority. The goal of all this pharmacy fraternity of any Indian nation is providing a best pharmaceutical care to the public. So I am happy to be here today among you here in International Conference on Emerging Trends in Personalized Drug Discoveries and Futuristic Approach. I feel this particular international conference quite aligning with the goal, providing that is the best pharmaceutical care to the public. The pharmacist, to attain this goal, not only should have the regular skills, they should attain some futuristic skills also. So I say our pharmacists should be future ready to attain this goal. And as clearly mentioned, many people before me mentioned, the very emerging trend now is personalized drug regime. As you all know, as of now, the present drug pharmaceutical regime is the generic one. That means if I get a fever, I'll get paracetamol. Whoever gets a fever will get paracetamol. It is a generic pharmaceutical regime. But now, the future is personalized medicines. These personalized medicines depends upon your genetic makeup, your lifestyle, your morbidities, the environment, the comorbidities, the environment, so that the patient receives targeted drug delivery and less side effects. Nowadays, as you all know, whenever sometimes I go out of Hyderabad airport, I always see there is a company, will be one booth I can see, they'll screen, they will scan, that means they'll sequence your genome and give the human genome. That means now the technology is developed. You just give a drop of your blood, they'll isolate the DNA. they will scan or sequence the whole genome and keep it with you. That will be a blueprint for your future. That will be a blueprint for your personalized medicine in the future, in the future. And the next one that is going to develop in the field of pharmacy is that is target-based drug discovery, which is really a favorite topic to me. The reason is I became sometimes nostalgic. I almost worked 7 years on my research on this, especially the drug-based drug discovery only, with the help of NIMHANS in Bangalore, with the help of the Saha Institute of Nuclear Physics, Kolkata. I'm rather like to bring little bit of my PhD topic, little synopsis, because it may be boring to you, but I'm little excited to bring it to the before this August audience. My topic was P-glycoprotein molecular dynamic P-glycoprotein, the structural changes, the nucleotide binding domain structural changes of P-glycoprotein. And it was an exciting topic for me. The reason is P-glycoprotein is an active transport. Such transports are involving in multi-drug resistance, especially in drug resistance. And it is causing drug resistance in chemotherapy of cancers also. So with the help of NIMHANS as well as with the help of science of nuclear physics, I designed the drug target, computer-based drug target by using modular. I docked by using AutoDock. I used GROMACS to simulate the molecular dynamic simulations. And finally, whatever the work I got it, I could not publish it. It is an anticlimax. The reason is everything is in silico models. I didn't use the X-ray crystallography structure. Keeping that apart, because I was excited, because I became nostalgic because of this by attending this conference. So keeping this apart, but for providing better pharmaceutical care to the public, I like to bring to the before this August audience what is happened to keep up this goal of providing better pharmaceutical care. PCI is taking so many measures under the dynamic leadership of Dr. Montu Patel ji. PCI recently undertook under PMKEY, LSSDC is conducting skill improvement program for our retail pharmacists. For that, already 15 centers have been shortlisted, and I should bring it to the notice of President Sir Montu Patel ji that one college, that is Koringa College, already completed the batch, and then they attempted the exam, final exam also, the 100% results, they passed with 100% results. This is the very first batch from our state, sir, completed. I'm thankful and grateful to you for giving this opportunity for our state to upskill the skills of our retail pharmacists. At the outset, I'm thankful for the chief patron, Laura Taigaru, then patron, Honorable MP, sir, other management members of Vignan College of Pharmacy, principal, Dr. Srinivas Babu Garu, Dr. Saujanya and other staff for inviting me for this function and for your wonderful hospitality. I deeply indebted to Dr. Montu Patel ji for, despite of his busy schedule, accepting our invitation and gracing us and visiting us in our state. And I am thankful to Dr. Venkat Ramana for coming all the way from Hyderabad and gracing us this occasion. Thank you, one and all. Thank you for giving this opportunity.
Thank you, sir, for your invaluable address. I am honored to invite Dr. M. Venkatramana, sir, member, Central Council PCI, to enlighten us by addressing the gathering. Sir, please.
Good afternoon to beloved President, Dr. Montu Kumar Patelji, Pharmacy Council of India, Dr. Lau Ratte garu, Chairman, Vignan Group of Institutions, my council colleagues, Dr. Radhakrishnamurthy garu, Dr. Prasad Reddy garu, Dr. Niranjan Babu garu, William Carey garu, Registrar AP State Pharmacy Council, and great and eminent speakers on the dais, Dr. Srinivas Babu garu, Principal, Vignan College of Pharmacy, Dr. Saujanya garu, Coordinator of this 3 days international conference, respected management members, Rangaraj garu, Satish garu, Padmav Reddy garu, Bojraj garu, and all senior principals from various colleges, senior faculty, teaching and non-teaching staff, Vignan Group of Institution, delegates, my dear and dear students, good afternoon to all of you. It gives me immense pleasure to be a part of this 3 days international conference at Vignan University. Really, it is very needy topic. Emerging trends in personalized medication. It is a really upcoming futuristic pharmacy profession, which is much more important. Of course, this topic might be new to our young undergraduate students because they might have not underwent this in their curriculum. This is a problem with our young people. Beyond your curriculum, you need to update yourself. As all of us knew that there are multiple opportunities, avenues to learn or update our knowledge. You need not to wait to see that in our curriculum. Rightly mentioned by our Radhakrishnamurthy garu, already the Pharmacy Council of India started working to update this curriculum in par with our current requirements. As you rightly mentioned that first 2 years will be the basics and next 2 years will be the specialized program in our pharmacy education. Our Honorable President is seriously insisting the Education Regulation Chairman to come up with a new syllabus which is in par with our current requirements. No doubt that future is our pharmacists, and in future the personalized medication will be flourishing like to maximum extents, because as all of us knew that as of now the dose calculation mechanism is not happening, so the dose is uniform for everybody. So in some of the cases these problems has been faced. So the personalized medication is much more required and it is needed for treatment of a kind of diseases. Apart from this, I hope regarding this current title, the speakers will be highlighting much more on this. I request all the young minds to participate and to show much more interest to update your knowledge and to utilize their session for your future. Coming towards our Pharmacy Council of India, just I want to brief few of achievements in our Pharmacy Council under the leadership of youngest President Dr. Montu Kumar Patel. As all of my teachers knew that Pharmacy Council has started in a very small building where even you cannot walk in the office. After that, Dr. B. Suresh has been shifted to a new building where at least we have a space to walk. Now, under the leadership, under the visionary leader, Dr. Montu Kumar Patel, now he has constructed a floor of Pharmacy Council with almost 40,000 square feet. And really it is just like a 5-star facility. And everybody has questioned when he brought this proposal. They questioned that already we have a council, but why we are planning for one more council by spending a lot of money? He said that, sir, it is a question of respect of our profession. It is not the question of money. So this is his vision, because getting a office, even NMC, you knew that NMC is the richest council in our this thing. Even NMC does not have such a excellent office. So I request all the principals or managements, when you find time, you please, when you visit Delhi, you please come to our office and visit the office. There you can see the vision of our dynamic leader, Dr. Montu Patel ji. Apart from this, he has come up with a lot of revolutions in our pharmacy profession, in education system, as well as quality of education, as well as infrastructure wise, as well as research side. As you knew that hardly it has been past 2 years after taking his charge. Most of them are putting a lot of pressure. What he did in past these 1 or 2 years. Really it is very much interesting to know that he has brought approval regarding the exit examination, which was pending from past 10 years. It has been done only with his involvement and with his interest on the profession. Second thing is GPAT scholarships. He said that there were a lot of discussions going on regarding these GPAT scholarships. Few of them said that, sir, let us reduce the money, yes, because ACT is getting the money from MHRD, whereas we are providing with our own funds. So let us reduce this GPAT scholarship. But he said, sir, our scholarship should be at least 1,000 more than the ACT. So this is his commitment towards our profession. In the same way, with a lot of his efforts, he went personally to ACT office. He went personally by holding the file to various bureaucrats and he made it to get approved. And now he has released the circular that now the students can apply for the GPAT scholarships. Not only for the current year, even for the previous year students also we are providing the GPAT scholarships. This is a commitment of Dr. Montu Patel towards our profession. Next thing is coming towards our 1:20 ratio. He knew that there are some kind of malpractices happening at the ground level. So he want to control these malpractices because major these malpractices are happening at the entry levels. So he said that let us control the number of teachers, and he implemented this biometric attendance system to protect all his existing teachers. Few of the teachers has passed various comments in the various groups that the teachers are losing their opportunities. But the quality of teacher is increasing. He want to see his teacher in a more respectable position. Because in few of the states, teachers are getting hardly 10,000, 15,000. He feel very bad about such type of instance. He always says that he want to see our teacher is highly respected in the profession. So this is his commitment. And apart from this, he has allocated various funds for the research activities. So we are waiting for final approval from the ministry. So I think shortly, in a couple of months or within this year, if so, he is going to come up with the grants to all our teachers and young scientists. And finally, he has allocated funds almost 20 crore rupees to upgrade the skills of our pharmacists and our teachers. So this is the commitment of Dr. Montu Patel towards our pharmacy profession, and this all has not happened it has happened in a span of only 2 years or less than 2 years. So full time he is working as an employee, and parallelly he is uplifting this quality of education and the standards of the council and is moving forward. So I request all the managements as well as teachers, please join with us and please support our pharmacy council, because we all are together, we all are traveling in the same boat. And very unfortunate thing is, I'm very open to say that in spite of his support to our pharmacy profession, our AP as well as Telangana management has filed case against our pharmacy council. I felt very bad because whenever I put any request to him on behalf of our colleges, he never says no. When he is giving such a positive response, you see, at each and every level, there might be some kind of difference of opinions. So there are various platforms to sit and to discuss and to sort out the issues. So I request at least hereafter, please don't file any such type of cases against the council because it will discourage us and it gives a wrong message from our states. This was my small submission to the managements. Hope that hereafter we'll be together and we'll work together. And finally, I thank Dr. Srinivas Babu be- because he asked me that, sir, I need President sir to join in online at least. Montu Patel sir has accepted to come only through online. But I insisted him that, sir, Vignan is one of the best institute in our state of Andhra Pradesh. You need to come personally and you have to visit our quality of education institutions in our state. So with my request, in spite of his busy schedule, he accepted and he came over here to visit our Vignan group of institutions. Thank you, sir. Whenever I put any request on behalf of Telangana as well as Andhra Pradesh, he never says no because he knew how best we are contributing in the quality of education. So thank you once again, sir. And finally, I thank the coordinator of this program, Dr. Sojanya Madam, for providing an excellent hospitality as well as inviting me for this very much important international conference. Thank you once again for all the organizers.
Thank you, sir, for your words of wisdom. I now request our beloved chairman, Dr. Lav Rataya, sir, Vignan Group of Institutions, on to address the gathering. Sir, please.
Respected chief guest, Mount Kumar M. Patel ji, and the respected members of the Central Council of Pharmacy Council, and the other guests on the dais, the guests of the dais, many principals and professors have come from different colleges, and my dear students, good afternoon, everybody. So, yes. I really congratulate my principal and other organizers and also the guests for attending this conference because without the guests, without the participants, the conference cannot be a successful thing. I really congratulate them for organizing this event. And with all the good wishes and best wishes of all of you here, other principals also, and the students and other people, this college has achieved the top position in India in the NAC score. Yes, though it is the first cycle. Of course, autonomy and all these things happens naturally, but in the NAC score, first cycle itself, it has scored the highest position in the country. So I really, and with all with good wishes of all of us, neighboring colleges also, they have extended the good wishes. I thank everybody over here. Because here Mount Kumarji Patelji is here. As a chairman of the institution, I have some concerns. Many things are happening, as just now Ms. Venkatraman is expressing in the last 2 years. It's good, wonderful. Without the good infrastructure of the office, you can't do anything. You can't create an environment and all that. That's wonderful thing. Many changes, teacher, faculty, student ratio, all these things, changes were happened. The other 3-4 things from my mind, number one is all the time we are speaking about this system, but I request the president to make the syllabus more flexible, at least to the autonomous colleges, so that to accommodate the other things very comfortably. And especially the Indian knowledge system, Indian systems of medicine are also working very well. You have totally neglected that system. It was proved, Ayurvedic, Unani and other systems are proved to be good. And we have a lot of evidences for that. So we have to accommodate all those things into curriculum, at least on the discretion, discretion of the colleges, you can make it happen. So, I mean, it will have a great impact on the society. This is number one. Number two is whatever we are claiming, the research potential, I mean, not the potential, research activity of the pharmacy colleges are at very low level. It is not happening. So at the level of faculty and also at the level of students. So it needs to be greatly improved. So higher education means school doesn't need a research at all. But higher education institution means first it should be recognized as the research, for the research, not for education. Education is the secondary thing. Making them pass the examination is the very third thing. The first thing for any higher education is the research. For any university or for any higher education college, the first thing is, I mean, inculcate, need not be big researchers. At least we have to inculcate the spirit of research among the students and faculty. Somewhere you have to make it mandatory, at least two papers for faculty kind of thing. Even the students are also capable of writing the papers. We can insist on that and make them reliable. This is so important. Everybody knows now the Hyderabad is the biggest center of pharmacy companies. Biggest center of the entire country. It is one of the biggest centers of pharmacy. Where it has come, everybody forgot. It has come from the research of IDPL. Without IDPL, no pharmacy companies in Hyderabad. Yes, it has come from the IDPL research. So that is the very important component in the colleges. So that is the most, I mean, a kind of spirit need to be encouraged, need to be installed among the students and faculty, and there must be some incentives. Maybe Pharmacy Council can do that. So the big research, when there is going on, it can be. And this research, colleges cannot do on its own. Research may be given by the Pharmacy Council also, like ASCT and UGC as funds. Research may be done through DST or maybe through some industry. So this kind of facility council need to be done with another kind of conference with the industry and other people. College alone cannot do it, cannot fund it, cannot equip, I mean, get ready with equipments. All these things must be done with the support of Pharmacy Council and with the support of industry and also with the college. The third the level is the college. This is the most essential part to make everybody is talking to make them futuristic pharmacists. How can we make them futuristic pharmacists? When there is a research involved into it, then only we'll make them futuristic pharmacists. If we make them pass the examination or giving a certificate, they cannot they cannot be future pharmacists. Yes. So that is a kind of, in the research also, a kind of tangential thinking must be inculcated. One of my students, I think he is very much there here, he is doing the research on the cancer. He says he is trying to prove that for cancer, it is not the habits of smoking or the food or other things are responsible. For cancer, he is trying to prove it is the thinking process that makes the cancer. He is trying to prove. He has written the papers in the Nature, elsewhere, and all these things. And the Boston University is working with him. And MIT University of America is working with him. Yes. I asked him, how can you do it? He has taken the sample of 400 people without any habits but affected by cancer. He is making with his supercomputer, he is very much here, doing it that. Yes, that is a tangential thing, very different thinking. Yeah, very different thinking. Ah, he is very much there, I'll call him. So, this is how, yeah, we can, any student can do that, can think. Any faculty member can think very tiny. Every doctor is thinking out of your smoking, out of your food habit, out of stress, out of these things. But he is trying to prove this. Boston University is working with him. He is very much in our university and working. And MIT is working with him. Yes. So this is how research matters. The other thing what I request the chairman Mount Kumar Patel is, so this see this the whole topic is emerging trends in personalized drug discovery. How you make it personalized if you do not know or aware of the IT knowledge. Yes, there is nothing in the syllabus, in the curriculum given by the Pharmacy Council, IT knowledge. There are no courses for IT knowledge. How can they make it a personalized drug? It is a data to be collected. It is data to be analyzed. It is data to be, the drug to be administered through that. Suppose if there are 10,000 patients, 10,000 patients' history need to be stored, need to be analyzed, need to be, then only drug can be administered. It all requires IT, digitalized knowledge, IT knowledge. There is nothing in that in our curriculum. So we have to introduce at least 7, 8 courses of IT knowledge to that. The future is pharmacy students. Future is once we are, we in the next 4, 5 years, if we get AI doctors, artificial intelligent doctors, they are very fast. Once if the drug is there in US today and tomorrow it would be there in India because AI doctors are connected. Now the present doctors are not connected to one another. If a drug is there today, it takes 1 year to come to India and administer. It will take another 6 months and test it for another 1 year. But it won't be there in the next 4, 5 years. Once AI doctors are in the field already there, so they will be totally, most of the doctors would be AI doctors, and the AI doctors, the biggest advantage of AI doctors is they are connected. The computers are connected world over, like our internet. This is what is going to happen to tackle all these issues or to make them future pharmacies, really future-ready pharmacies. So we have to, yes, IT skills need to be given to them, need to be introduced in the curriculum. Otherwise, they will be irrelevant, very, very irrelevant. So nobody bothers about the regular practice. All the curriculum was designed 25, 30 years back. It should be revamped. That is what I was sharing with the President also. It needs a big revamping at this phase because we have to revamp like how the communications are revamped with the cell phone, how the transport is revamped, how the whole administration is, Andhra Pradesh is introducing WhatsApp governance. WhatsApp governance. You need not go to office, you need not request anybody, you need not beg anybody, you need not corrupt anybody. You can do everything in WhatsApp. When you don't react to that, when you don't react to the trends, you are nowhere. You will be irrelevant. So you will be, the students would be irrelevant. You don't update yourself. First, principals and faculty need to update and introduce the same thing or make the students ready for that. Then only they will be they will be, good in this country and also any other country. That's it. These are the 3 requests. Again, I am repeating. The first thing is Indian knowledge system is a great knowledge. We can't afford to ignore that. Somewhere we have to give the space for that. That is the first thing, yes, especially in the health systems, Indian knowledge is great. In other systems also, okay. Architecture, we have seen many temples, how they've done. It's a great knowledge. So in the same way, pharmacy also need to introduce Indian knowledge systems into the curriculum. The second thing, research should be considered as the main thing. I don't know how they make it a policy because the council members are there, 3 members and chairman is very much there. We have to make it a policy. What is it you are expecting, minimum things expected from a college, from a professor? How many papers are expected? What is the kind of H index from a professor you are expecting? This kind of regulations, policy making, one can do that. And third thing is, we have to make the students to be aware of, to be familiar with IT knowledge. Otherwise, they would be totally irrelevant. Once again, I thank the President of Pharmacy Council, Montkumar Patelji, for his great presence, inspiring presence over here, and other guests, Radhakrishna Garu and other members of the council, registrar, everybody over here. I thank you, everybody, once again. Thank you very much.
Thank you, sir. You were a true source of inspiration to all of us. It's our privilege and honor to invite Dr. Montu Kumar Yam Patel, sir, President, Pharmacy Council of India, to address the gathering. So please.
Thank you.
Namaskar. Good afternoon. Vigyan यूनिवर्सिटी की कॉन्फ्रेंस इमर्जिंग ट्रेंड इन पर्सनलाइज्ड ड्रग डिस्कवरी फॉर फ्यूचरिस्टिक अप्रोच organized by the Vigyan University. The chairman of the Vigyan group of institution, Sri Dr. लव रते और हमारे एग्जीक्यूटिव मेंबर डॉक्टर वेंकट रमना जी, सेंट्रल काउंसिल मेंबर SLN प्रसाद, डॉक्टर निरंजन बाबू, and most senior respected member Sri Radhakrishna, Dr. Radhakrishna Murthy ji. Andhra Pradesh Pharmacy Council Registrar Dr. M. William Carey and Dr. P. Sivanas Babu, Principal of the Vigyan Pharmacy College, and respected all the principals come from the Andhra Pradesh, and the dear students and the dear eminent speakers who are come from the different part of the India and the abroad. Many discussion and the speech given by the Honorable PCA members and the chairman of the Vigyan group of the institution. But you know, from Pharmacy Act is established from the 1948. Now 75 years we are completed. And in Corona period, we are become a pharmacy of the world. If our education system is not working, if proper pharmacy education not taken by the good institution and the good teachers, then India never became a pharmacy of the world. So it is all credit goes to the good institution and all the teachers who are researchers and the scientists who are created the good industrialist and the good good quality pharmacies in for the Indian people, also the other country. Now our Prime Minister vision for the how can we make a pharmacy make a globally, because already the pharmacy of the world, but how can we supply our manpower, because we have a skills, we have a good knowledge, we have a good infrastructure, we have a good education system, and how can we give a service to other country, because Vasudev Kutumbakam, you know the Vasudev Kutumbakam. So we have to work for not for the India, we have to work for the other country also. So our Prime Minister vision, how can we make a good doctors, pharmacists, nurse, and the 56 are the paramedical courses, lab technicians, and the other things. So in future, I think good quality pharmacists, nurses, doctors are working already working in the other country, but proper system by the government, other countries and the Indian government make a bilateral MOU with the other countries, and the proper channel will be set up by our central government. And in future, we can easily go other country and other country people also work in the India also, because all the manufacturing plant, production plant, and the API industry and the medical device industry in the India. So other country people are also want to come here and the work for the industry. So government are planning easy to easy visa for the our students also, and their other country people also work here easily. So it is our Prime Minister vision. Accordingly, the Pharmacy Council India is working, because already when I took a charge in the Pharmacy Council India as a president, so many challenges, because I am not an academician, I am only administrator, because I am also the president of the Gujarat State Pharmacy Council for 7 years. So I all because I observe many things from the ground reality, because when the I am also facing a registration, because basic things of the students for the registration, not after graduation, after taking a degree of diploma degree, first they have to face the challenges for the registration. All teachers are also facing the problem for the registration renewals, and it is a primary requirement of our profession. But the all the state pharmacy councils are working independently, because they are not knowing the government powers and the public powers, and they are always work for the elected member, nominated members, and who are many people's. Excuse me, don't talk. Many people are in state pharmacy council working for own agenda. So when I took a charge in the Gujarat State Pharmacy Council, because I are also facing a problem of the registration. So first, only I taken a one agenda. How can become a paperless council, and how can students get a easily registration system and the one click renewal registration, because in Gujarat and in every state, I think many the office is in one place, central place, and the distance from the central place is very far away, at least 100 kilometer, 200 kilometers. So students has to travel for the renewal, registration, and the many things of the state pharmacy council, because in career every state you have to require of the state pharmacy council, and the many issues are also faces by the pharmacy. So in Gujarat, I started through paperless council, and I think a single click our register can renew. When you apply online, you have to pay the online payment, and the single click you get a registration and the renewal. So in Gujarat, from the India, first became a paperless council, and from there work government are also recognized, and the government of India also recognize our work, and many other councils same follow for the paperless council. But what I say, the problem for students first is registration. Also, I observe many things. So from there, now the Pharmacy Council India, as a president, I received many mails from the students and the teachers, because state pharmacy council are not easily transferring the registration, and also not get a registration in a time. So in Pharmacy Council India, I started a portal for the students portal and the faculty portal. In students portal, from last year we are started the student portal. All students get to register in the portal. When they are pass out, only they have to single click whatever, matlab, whichever you council, Gujarat, Maharashtra, Andhra, Telangana, whatever you council, you have to get registration. You have to just click on the State Pharmacy Council name, and your all document will be shown to the State Pharmacy Council. Your application also shown to the Pharmacy Council, and the register of the any State Pharmacy Council, they have to give registration within a 24 to 48 hours. This system will be established by the Pharmacy Council India. Second, we are also, because teachers are working many years, many senior teachers are working many years, but they are not getting a justice, and some good institution are giving a good salary, also give additional benefit, but most of the above 50% institution I observe, they are not recognize our teacher service. They are only not respect our teachers and give the salary, and one hand they give the salary, second hand they withdraw the salary from the account. This is the reality of the now pharmacy education. So Pharmacy Council India started the biometric attendance Aadhaar link system, because in India first time we are started in the higher education. For the already medical council are working, but they have 500 institution medical college now, including government, maximum 700 institution in India. But in Pharmacy Council, more than 7,000 pharmacy institution in India. So you imagine what scale we have to implement the system. Because in 700 easily we can implement, but in 7,000 institution, it is a challenging task to implement the biometric attendance system. And we have to also link up the Aadhaar with all the teachers. And second phase, we have to link up with the students' Aadhaar, because when the students are studying in the institution, because many deemed university or private university are giving a backdate degree, also non-attending degree. So these are all the people know from the pharmacy institution, and the good teachers and the students. Some university are running without attending and without teachers. So this is the solution we are came from the through IT for the good quality education system and the good future of the pharmacists. So we are linked up with the all Aadhaar teachers, first phase teachers. From after 1 or 2 months, we are linked with the students also. So all the data will be reflected on the Pharmacy Council India portal, and from their portal, because Government of India started the healthcare professional registration system, HPR system. Doctor are already enrolled in the system, nurses are already enrolled in the system, but pharmacists till date not enrolled in the healthcare professional registration system. So PCI, I think very soon we will start HPR system. So all the pharmacists can register on the portal for the healthcare as a healthcare professional, because when the pandemic situation or what in emergency situation, government can access to government have access for the healthcare professional teams. So any pandemic situation, government can create a workforce for the emergency situation. So these are the vision of our Prime Minister, because in Corona many challenges faced by the government of India for the find out the doctors, nurse, pharmacists, and the very healthcare even lab technicians. So now the government of India also are technically and IT, they are created in the healthcare system, as like the chairman of the Vigyan institution said, not we are not included the IT in our education system, but in new syllabus, because already the syllabus almost final, because we before 2 months the one program is also in the Hyderabad for the finalizing of the syllabus. So in new syllabus, the specialization from the BPharm level and the MPharm level, super specialization, and the IT related, AI related software system, because now all the manufacturing plant of the pharma industry are going to the automation with the support of the AI. So pharmacist has to upgrade his skill for the technology, because we are know the mobile technology, but in software technology is different, because if our students from the diploma or degree, at least he sit in the medical store or one pharmacy store, he cannot run a software for the sell purchase bills software. At least basic training you have to give, because it is a basic training, because at least pharmacists know the what is the software in the pharma industry to up to retail, because it is all the link up system from the manufacturing to the customer. So we have to at least know how can use our day-to-day softwares, what is the technology, how can you, because now the online pharmacy are day by day increasing, even Pharmacy Council in India are opposing the online pharmacy, but in the IT Act they are getting a benefit and they are running the online pharmacy, because the online pharmacy are very dangerous for the pharmacy field. The our in one shop at least they have to require one pharmacist. If the in online, if they get a one license for the Andhra Pradesh, and and below the they can hire the arts, commerce, BA, BCom graduate for the delivering of the medicines, for the picking of the medicine and dispatching of the medicine. So from one online pharmacy can lose our job opportunity from the different pharmacy stores. So it is a very dangerous for us. Already PCI taken a stand for against to him, but it is not a technology means online pharmacy, but technology means manufacturing medical device, because no all the medical device, because after the corona period we are importing the good medical device in the corona period, but after the corona government given the 1000 crore for the medical device park, 1000 crore for the API park. I think in Hyderabad, Telangana, one API park and medical device park already established. In Gujarat also medical device park is established. In Noida also medical device park is established. So many opportunity for the students, but we are only thinking for the manufacturing of the medicine, tablet, capsule, injectable. But despite of this, medical device is a big industry. We have to focus. We have to think for the medical devices opportunity, and another cosmetic industry and the nutraceutical industry, because we are never think about the cosmetic industry and the nutraceutical industry, because the every day every family consumes one or two multivitamin or multivitamin syrup or multivitamin tablet for the nutritional, as a nutritional, because in food the we are not taking a healthy food, and the very busy and hectic life schedule. So everybody needs to the multivitamins and all the things. So we have also focused the nutraceutical industry. It is a big industry, more than the pharma industry. Nowadays, the nutraceutical industry values is more than the pharma industry. So you have to focus on the nutraceutical industry, cosmetic industry. You all know the many cosmetics, because in laboratory we are preparing the lab scale cosmetics, but in reality I think no, many less pharmacists are working in the cosmetic industry. Means till date I never meet the any pharmacists who are working in the cosmetic industry. Yeah, they are working in the manufacturing, but they are not in the marketing, selling, or any other medical cosmetic industry, because cosmetic government of India also notified the new regulation, and they are also compulsory to as a competent person as a pharmacist, because earlier there is no requirement of the pharmacy. Now the government of India are as a competent person you have to hire pharmacist also. So many things government of India updated, but still we are not thinking for the cosmetic nutraceutical industry, even veterinary industry. You know India is a very big dairy productions and the very huge the farming, but we are not focusing on the veterinary industry. Veterinary industry are also big, but our pharmacists are never working for the pharma, only veterinary medicines or veterinary medicine manufacturing. So many opportunity students we have, but we are not grabbing. Only BSc and the other people, they are working in this industry and we are losing our respect and we are losing our also losing our professional. So these are the new, but in new syllabus, this all area we are covering. That's why I updated you. We are covering all the area we are remaining from the many years. So we are take all the area in the new syllabus, and new syllabus almost final, but we have to approve from the government of India maybe next year, within a 1 year, means next year we will implement the new syllabus also. Another the skills, because government of India also, our prime minister also focusing on the skills, because degree, everybody can degree easily, take a degree easily, but without skill you cannot survive in the competition. So how can upgrade our skill? Already government of India allocated for CR for the southern region, for Andhra, Telangana, Karnataka, Tamil Nadu, and the Lakshadweep, all the 6 states, we allocated for CR for the training of the pharmacist. After graduation, the pharmacists need the pharmacovigilance skills, communication skills, software skills, and many skills you have to require. Whatever in the not what daily wages you have to work with the industry and the in the pharma store. So you need a skills. So government of India planning to train 24,000 pharmacists in the southern region. But despite of this, Pharmacy Council of India allocated 20 CR fund for the scaling of the pharmacies. We are developing the center of excellence, means all the lab scale manufacturing facility in the poor part of the India, and you have to get a training from them how can run an instrument, how can use a software, how can you do the communication with the public. Different type of program we are established in future. So we have already working on it, and maybe very soon we will finalize the center of excellence. One most challenging thing is GPAT. You know the GPAT, because students daily wages 1,000, minimum 1,000 missed call. Students are intentionally calling me for the irritating the president and the, so I block every time, every day 200 call in my phone. But finally, because the students are the not, because they are facing challenges, that's why they are calling me. But I understand, but this, the situation is not in my control. We have to take approval from the councils, ministry, and the many layers for the approvals. So finally, from the support of the, from the Pharmacy Council India members, we are coming out the solution for the GPAT fund, because we are changing name Pradhan Mantri PG Study Scheme Yojana. So we are also change the name of the GPAT. Now the scheme name also change. But PCI is only 130 CR fund when I took a charge. And how can we give a stipend to the student? So it is challenges, but we are increase the PERC free. You know the or the South people. Nay, it is because all the management people are against me all over the India. They are making a group. How can fight with the PCI? Because they know the old PCI. Because they are knowing the old PCI. Only take the fund, not give the fund to the people. So I have we have to change PCI image also. Now the GPAT 300 crore fund we have to release within a 1 year. So the big challenge is what PCI, but we are converted to the challenges in the public benefit. So now 300 CR we are allocated fund for the 3 year, three batches to give the GPET stipend. So this is a big achievement with the Pharmacy Council India. And 130 CR office we are purchased because the day by day 7,000 institutions are working. So we have to deliver service because no management, teacher or student suffering from the due to the system of the Pharmacy Council of India. So we are also planning to hire 150 new staff for the Pharmacy Council India, and we give the service because we are the facilitator. So always Pharmacy Council India give the service to the all the stakeholders, students and the teachers because we are not sit for the harassing the people. So that's why we are the facilitator and that's why we make a system. How can we facilitate our pharmacists, teachers and the management for the better service? So this is a new office. We are already make a high tech office for the also make a 150 150 people training center also because we made a one conference room because every month we have to train teachers and the registered pharmacists and also some PG students. So 140 to 150 teacher or students every month PCI can give the training by own building, and also we have a cafeteria of the 150%. So permanently one in-house setup given by the Pharmacy Council India for the training teachers, students, and the good conference at the Delhi level. So you have to take benefit because we can charge from the people for the conference to use our facility. So if you are interested to give any conference to Delhi, so we provide you place and the restaurant, and you have to pay the PCI. But from the Pharmacy Council India, we can give a free of cost to our pharmacist. But as a professional, if pharma company wants to 1 day meeting, then we can give it on the charges basis. So new changes are always welcome by the stakeholders, but many things we have to discuss, we have to face. Also, many things we have to face is challenge in future from your side, and you are also facing problem from our side because biometric and other things are not good for the management. But you have to implement it. Otherwise, the approval process in your approval, we will hold the approval of all the pharmacy institution. So from this system is implemented from this year. So we are already circulated within a 1 month you have to install. Otherwise, we will hold your approval for this year. Otherwise, you have to apply for the next year. So without facing any legal challenges from the PCI, you have to apply and you have to implement the system. Another exit exam because the draft already made before I took a charge, but I finally stayed for the approval in the ministry. So from many years, it is not approved by the any ministry. So from our team, we make an effort and draft is approved. But the problem when the policy will publish by the government, open for the public comment. That's why we are not taking it seriously. And 3 paper for the 3 hours for the diploma student, it is not possible. Practically, it is not possible. So we are changing for the 1 paper for 3 hours, and also because 3 paper cost will be increased. It is also given the CSR fund by the government of India to reducing fee. Otherwise, it is a 15,000, but up to the 6,000, the given the government of India given the CSR fund for the diploma student for the reducing the fees. But now I think 1,500 or 2,000. Now the new exam will be implemented by the government of India. And the notification already in the process for the 1 paper or 3 hours for the diploma student. So it will be implemented. But the during we have to face many represent from the students, teachers for the registration of the pharmacists because they are pass out. Exam is not conducted by the Pharmacy Council of India. It is not a problem of the student side, but it is problem for the authority side. So we given the 1 year registration for the 1 year temporary registration. But after 1 year, you have to clear exam and you have to renew your certificate. So we are always come out from the solution. How can we help the students, teachers, and the management? Many new policies for the mode drop policy, what the AICT and UGC, whatever in past giving the benefit to the pharmacy institution and the teachers, all policies will be implemented by the Pharmacy Council of India. Already budget are also allocated, but IT system we have to develop. So first GPAT already we are clear. Many issues are coming, but now within a 2 3 months, we hopefully will implement all the policy for the teachers training, student training, mode drop policy. Many policy we are implementing for the benefit of the pharmacy professional. And the thank you for the inviting me in the inauguration of the Vigyan College of Pharmacy and giving the opportunity to interact with you. Thank you to all management, teachers, and the students. Thank you. Jai Hind, Jai Bharat.
Thank you, sir, for your great motivation. We request our dignitaries to release the souvenir copy of our international conference on emerging trends in personalized drug discoveries, a futuristic approach. I request convener, organizing secretary, co-organizing secretary, and Dr. P. Ravishankar, sir, onto the stage for souvenir release. Indeed, it's our prime concern to felicitate the delegates who have spared their valuable time to enlighten us on this auspicious occasion. I now request the dignitaries on the dais to honor Chief Guest of the Day, Dr. Montu Kumar M. Patel, sir, PCI President. I request dignitaries on the dais to felicitate our Chief Guest. I request our supporting partners, Dr. Jitendra Chimakurti, Dr. Y. Srinivas Rao, Dr. M. Srinivas Murthy to join the felicitation. Sir, please. We request our supporting partners to join the stage. I now invite Dr. M. Venkatramana, sir, to get felicitated onto the dais. I request the delegates to honor Dr. M. Radhakrishna sir. Let me now request the delegates to felicitate Dr. S.L.N. Prasad Reddy, sir. May I now request the delegates to felicitate Dr. M. Niranjan Babu, sir. I now request the dignitaries on the dais to felicitate Dr. M. William Carey, sir. I now request the dignitaries on the dais to felicitate Dr. Srinivas Mutalik, sir. I now request the delegates to felicitate Professor Javid Ali, sir. Now I request the delegates to felicitate Dr. Chandra Godugu, sir. Now I request the dignitaries on the dais to felicitate Dr. Giribabu Nelli, sir. I now request the dignitaries on the dais to felicitate Sri Hanumant Rao, sir. I now request the dignitaries on the dais to felicitate the organizing members of this conference. I now request the dignitaries to felicitate co-organizing secretary Dr. J. Nagalakshmi ma'am, associate professor. I now request the dignitaries on the dais to felicitate organizing secretary Mr. P.V.V. Sivakrishna sir. I now request the dignitaries on the dais to felicitate convener of the conference Dr. P. Saujanya ma'am. Now request the dignitaries on the dais to felicitate co-patron of the conference, Dr. P. Srinivas Babu, principal. I request dignitaries on the dais to felicitate our supporting partners, Dr. Jitendra Chimakurti, head of the department, Vignan's Foundation for Science, Technology and Research. We now request the dignitaries on the dais to felicitate Dr. Y. Srinivas Rao, sir, head of the department, Vignan's Institute of Pharmaceutical Technology, Visakhapatnam. I request dignitaries on the dais to Dr. A. Pramila Rani, University College of Pharmaceutical Sciences, ANU. I now request Dr. A. Pramila Rani, ma'am, Principal, University College of Pharmaceutical Sciences, ANU, onto the dais to be felicitated. Dr. K. Venkatramana, Principal, ASN College of Pharmacy. Dr. A. Narendra Babu, Principal, Sir C.R. Reddy College of Pharmaceutical Sciences. Dr. V. Suresh Chennupati, Chennupati Indo-American School of Pharmacy. Dr. A. Venu, Daggubati Chenchuramaya Memorial Pharmacy. Dr. M.V. Nagabhushanam, Principal, Hindu College of Pharmacy. Dr. Bhavani, HOD, Department of Pharmaceutical Sciences, JNTUK. Dr. Suvarna Jyoti, Principal, Koringa College of Pharmacy. Dr. A. Sunita, Principal, KVSR Siddhartha College of Pharmacy. Dr. G. Sudhakar Rao, Principal, Malnane Lakshmaya College of Pharmacy. Dr. Manchineni Prasad Rao, Principal, Maulana Azad Memorial College of Pharmacy. Dr. Brahma Reddy, Principal, Nalanda Institute of Pharmaceutical Sciences. Dr. Brahma Reddy, Nalanda Institute of Pharmaceutical Sciences. Dr. A. Ravikumar, Nimra College of Pharmacy. Dr. J. N. Suresh Kumar, Nasropeta Institute of Pharmaceutical Sciences. Dr. Y. Ankama Chowdhury, NRI College of Pharmacy. Dr. Jagadish Panda, Principal, Raghu College of Pharmacy. Dr. Y. Padmanabha Reddy, Raghavendra Institute of Pharmaceutical Education and Research. Dr. C. Madhusudan Shetty, Shantiram College of Pharmacy. Dr. K. Srinatraj, Sri Vishnu College of Pharmacy. Dr. B. Tangabalan Sir, SIMS College of Pharmacy. Dr. D. Ranganaikulu Sir, Sri Padmavati School of Pharmacy. Dr. Rajkumar Sir, Principal, St. Mary's College of Pharmacy. Dr. Jeevan Manibabu Sir, St. Xavier Institute of Pharmacy. Dr. T. Vinay Kumar Sir, Victoria College of Pharmacy. Dr. A. Lakshman Rao Sir, Vallabneni Venkatadri Institute of Pharmaceutical Sciences. Dr. G. Sumalata Ma'am, Vikas Institute of Pharmaceutical Sciences. Dr. G. Ramna Reddy, Professor, KVSR Siddhartha College of Pharmacy. Dr. G. Madhu Sir, Principal, Kids College of Pharmacy for Women. Dr. V. Uma Maheshwar Rao Sir, Academic Dean, Raipur, Anantapur. Dr. Adimulapu Satish Sir, Chairman, Samuel George College of Pharmacy. Dr. Jupudi Rangaraju Sir, Chairman, Hindu College of Pharmacy. Dr. Bojraj Sir, Veda College of Pharmacy. Dr. M. Sunil Kumar Sir, Anjareddy Pharmacy College. Dr. N.S.P. Prasad Rao Sir, Mittaballi Pharmacy College. Dr. Harinatha Baba Sir, Krida College of Pharmacy. Dr. Bharati ma'am, KVSR Siddhartha Institute of Pharmaceutical Sciences. Dr. Srinivas sir, Srinivasa College of Pharmacy, Vijayanagaram. Shri Galla Durga Prasad sir, member AP Pharmacy Council. Sir, please. Request dignitaries on the dais to felicitate Chairperson of Morning Scientific Session, Dr. V. Sai Kishore, Head, Department of Pharmaceutics, Bapatla College of Pharmacy. Please. Request all the faculty members of Vignan Pharmacy College to join for a group photo. I request all the faculty of VPC and VFSTR to join stage for a group photo. I now request organizing secretary of this conference, Mr. PVV Sivakrishna sir, to propose formal vote of thanks.
Good afternoon, all of you. Distinguished guests, esteemed panelists, fellow delegates, and the incredible organizing team. It is with immense pleasure that I stand before you today to express our sincere appreciation for your participation and contribution to this remarkable conference. I wholeheartedly thank the participants for their thoughtful engagement throughout the conference. I express sincere thanks to all sponsors and partners for their generous support, so which enabled the conference to reach its full potential. I wholeheartedly thank my colleagues and my student friends for their full extended over a month in making this seminar a grand success. Thank you, one and all. Thank you very much.
Now I request VPC team for a dance performance, classical dance performance.
Dear students, the next scientific session will start at sharp 2:40 p.m. in this seminar hall by Professor Javed Ali sir. All the principals to have the lunch at Vignan Pharmacy College premises, and student delegates also at the premises of Vignan Pharmacy College. Faster.
Principals and delegates and
All the principals, faculty delegates, and student delegates.
I request all principals, faculty delegates, and student participants to have your lunch at Vignan Pharmacy College premises. I request the dignitaries to please be seated for 2 minutes for this performance. After this performance, you can have lunch at Vignan Pharmacy College principals.
వక్రతుండ మహాకాయ కోటి సూర్య సమప్రభ కురు సర్వకార్యేషు సర్వదా ఆ గణపతి గణపతి మనసా స్మరామి మహాగణపతి మనసా స్మరామి మహాగణపతి మనసా స్మరామి మహాగణపతి మనసా స్మరామి వశిష్ఠవామదేవాది వందిత గణపతి మనసా స్మరామి వశిష్ఠవామదేవాది వందిత గణపతి వసుతం దేవసుతం కోటి ప్రకాశం శాంతం మహాదేవసుతం కోటి ప్రకాశం శాంతం మహాకావ్య నాటకాది ప్రియం महाकाव्य नाटकादि प्रियं मूषिक वाहन मोदक प्रियं महाकाव्य नाटकादि प्रियं मूषिक वाहन मोदक प्रियं गणपति मनसा स्मरामि
did in silico study in order to find out how they're going to bind up with the target when both drugs are available. So in silico study talked about, yes, these can be combined together, and accordingly, they were tried in the breast cancer cell line in order to identify a ratio, a combinational index. We wanted to find formulation as such. Screening was done. Which was a best solid lipid, which is showing a high drug solubility in the solid lipid, the liquid lipid, and accordingly a binary mixture was used. And this binary mixture was further taken in terms of finding out the compatibility in terms of crystallinity index and so on by DSC. On the basis of this, we also try to find out the compatibility issues with drug and excipient interaction, and it found to be taken further as such, right? So this is an optimized formula. I've not shown the DOE curves and all because they must be very heavy for the audience also, so I did not incorporate. The formulation was characterized for particle size and for the surface topographic studies, right? And in vitro analysis were done in different media for the drug combination at 0.1 normal SCL, mimicking the upper GI SGF pH. pH 4.5 showing the proximal small intestine pH, and pH 6.8 showing the small intestinal pH as such. And accordingly, the assessment was done and the formulation was taken for the for comparative studies for in vitro data as such. The in vitro depth of permeation was also studied by confocal microscopy, and penetration was like, in the intestine within 2 hours, it was able to achieve in the plasma levels and so on. The various, as I mentioned already, the drug has been mixed with an phytoconstituent which was having antioxidant activity also. So we determine intracellular reactive oxygen species ROS and superoxidase dismutase assays was done. This was just to show, because in the breast cancer it is being reported that there is an oxidative stress which also lead to breast cancer cell development, and accordingly we wanted to assess the antioxidant activity as such, right? In vitro lipolysis was analyzed where the lipids in the formulation were degraded, and we just tried to find out what is the solubility and the non-soluble part within the NLC formulation. Pharmacokinetic studies revealed that there was a enhancement in the AUC values particularly, and showing a better relative bioavailability, and this was published in one of the good paper. And then we also do pharmacokinetic studies of ribociclib with GT and NLC, and then there was an enhancement in the bioavailabilities as such. This was also published in one journal of drug targeting, where a combination delivery of ribociclib and green tea extract was mediated as such, right? Histopathological analysis was done in order to establish acute toxicity studies, and the stability studies were also performed, because stability is an issue in case of the lipidic system, particularly liposomal preparation or solid lipid nanoparticle or whatever. So in vitro, in ex vivo and in vivo results showed a prominent potential for bioavailability enhancement of RBO and effective breast cancer treatment. In terms of one student is now taken this project further in order to make surface engineered folic acid conjugated folate receptors, for targeting folate receptor, formulation. The study demonstrated that NLC of RBO and their combinational approach is an extremely potential approach for enhancing the therapeutic prospect as such. So what we conclude out of this formulation, preparation methods and development methods so far, NLC is a potential approach for improving the oral viability of highly lipophilic drug with poor aqueous solubility. And I have mentioned already, it is going to bypass portal network. So accordingly, it is also going to bypass extensive first pass metabolism also. drugs which have affinity for PGP flux, they can also be, avoided by use of proper lipidic system, which have been reported to be the inhibitor for PGP flux as such. In a way higher caliber of NLC systems shadows the partition of other lipid and base dosage form. Disrupted matrices of NLC consequently lead to higher drug loading, higher drug entrapment, moderate drug release, and ultimately enhanced drug absorption as compared to the other lipid-based formulation. So the future prospect in this study is promising results obtained using NLC suggests that the formulation should be extensively tested in the human subjects as such. In vitro models, preclinical and clinical data on a wide range of drug candidates with different physiochemical properties would help elucidating the mechanism of action as such, and exploration of the potential of NLC formulation in improving the absorption of highly lipophilic drugs in conjugation with thorough understanding of the effect of formulation components on the flux transporter, metabolic enzymes, and lymphatic uptake would help in completing addressing all the negative aspects of oral drug delivery of lipophilic drugs in particular. So these are the references we have taken. These I acknowledge my colleague Dr. Sanjula Babuta, and these are all my doctoral students. Some of them are presently, continuing with their PhD program. These are informed students as such. These are our research group at Jamia Hamdard, and this is the group which are working on breast cancer, research and all. So thank you for your patient listening. Because of scarcities of time, I did not went into discussion and detail. Thank you for, and in case you have any query, I will be happy to answer, right? Thank you very much.
Thank you so much, sir. Thank you for a nice and useful for the current scenario. The session is very nice. Any queries from the audience? Sir, I have one question, sir. Sir, you mentioned that green tea extract can be used. How it works, sir?
Vaccines are being reported to be an antioxidant, and these specifically phytoconstituents are being reported to be having activity against breast cancer. There are a lot of it.
Okay.
So because of these two molecules, it's being used. That's why.
So is it stability regarding the stability, sir? Is it?
Actually, in the case, it is basically GTE in selection, but when we prepare it, we use the isolated catechins. So from Sigma, right?
Yes, sir. Okay. Thank you, sir. Okay, thank you, sir. Thank you for sparing your valuable time, and you come to the Vignan University from the New Delhi to here. Thank you so much for your valuable time. Thank you, sir.
Thank you, ma'am. I now request Dr. G. Chandraya, sir, Department of Regulatory Toxicology, NIPA, Hyderabad, Dr. Jitendra, sir, HOD, Department, Pharmaceutical Sciences, VFSTR, and other faculty delegates to felicitate Dr. Javed Ali, sir. I now request members to felicitate the chairperson, Dr. Bharti. I feel it an honor to introduce the chairperson, Dr. Midhun Rudrapal, sir. Dr. Midhun Rudrapal is Associate Professor at the Department of Pharmaceutical Sciences, School of Biotechnology and Pharmaceutical Sciences, Vignan Foundation for Science, Technology and Research. Dr. Rudrapal has actively engaged in teaching and research in the field of pharmaceutical and allied sciences for about 15 years. He has 200 plus publications in many peer-reviewed and scholar national or international journals and 20 plus edited or authored national and international books to his credits. He has filed and granted a number of Indian and international patents. He is a fellow and life member of various esteemed professional associations including ICS, IPA, APTI, ISCA, IPS, among others. He has been recognized as journal reviewer, journal guest editor, advisory board member, editorial book and book volume editor for several prestigious international publishers, including Elsevier, Wiley, Springer, T&F, and others. Dr. Rudrapal has been ranked the world's ranking of top 2% scientists by Stanford University, USA for the year 2023 and 2024. I now invite Sir to introduce the speaker of the session. Sir, please.
Good afternoon, everyone. So first of all, I would like to thank the organizing committee of Next Gen Pharma Connect 2K25 for giving me the opportunity to chair the session. So we have our next speaker, Dr. Chandraya Godugu from Naipur, Hyderabad, who is a distinguished academician and scientist in the field of pharmacy and pharmacology. So please join me in welcoming the speaker of the next scientific session. Will share his expertise with us today. It is truly an honor and privilege for me to introduce Dr. Chandraya Godugu. So he's a distinguished academician and researcher with 14 years of experience in pharmacological and toxicological research. His contributions include he has expertise in pharmacology, nanotoxicology, and drug delivery. He has 4.5 years of teaching and research experience at Naipur, Hyderabad. And he's a postdoctoral fellow at Florida A&M University under Professor Mandeep Singh Sachdeva. He has 44 publications to his credit. He has US patents. He's a receiver of DST DAT Fellowship. He is also an achiever of RV Patel Pharma Innova Award. He is a leader of robust research program supported by significant sponsored projects. He has contributed to various international committees and delivered several invited talks internationally. Today, the title of his talk is Opportunities and Challenges in Phyto-pharmaceutical Research. Now I welcome Sir to please come over the dais and please enlighten us with your talk. Thank you. Okay.
Thank you, sir, for the wonderful introduction. But I can see the resume you have taken was probably 7 8 years back resume. Unfortunately, I didn't update in my website. That was my mistake. So yeah, so I have a different credentials now, and yeah, that I don't mind it. So yeah, I will be more interacting with the students than the faculty. These are already already done with their learning. Of course, they still have to do the learning, but I will be more interacting with you people. So I want to be with you people, and you should ask me the questions, and I'll be also asking the questions, right? Yeah. So I'm a pharmacologist basically. I also work in drug delivery. So I feel if you combine pharmacology with the drug delivery, you can do differently than what just pharmaceutics person think, right? And also I have a diploma in toxicology, which is American Board of Toxicology. So what it tells is you may have to sometimes integrate and diverse from your area. That's where you will get more opportunities. Just only, of course, yeah, you should go more deeper as well into your subject. Sometimes it is also required for you to interdisciplinary, right? Either you go yourself or you go and collaborate with the people. That's where you get um, more opportunity to achieve the things, right? See my title, Phyto-pharmaceuticals. Rataya sir was also talking about today morning. Importance of the Indian traditional medicine, right? And if you see, we are slowly, and we already moved away from our rich source of herbal medicines. We are not taking the right vegetables. We are not eating the enough phytoconstituents through our food, and all the processed food already losing a lot of important the components which might have improved your health. Either to be improved, it can be immunity, or it can be also long-term chronic disease like diabetes. Why we are actually getting diabetes in so early age, and why so many increased cancer cases? Because of all the issues, right? And there are a lot of phytopharmaceuticals which can be actually protecting your body. So if we can actually, why we are not able to do it? Because we don't have a lot of scientific validation for the plants we have. One of the plant I have taken for last 10 years, and I could see a lot of potential with one plant, which is a neem tree. And there is a chemical called nimbolite. So I also work with ashwagandha, which is in Vidafernie. You all might have gone through the pharmacognosy textbooks. Vidafernie is one of the phytoconstant. It's an excellent anti-cancer agent as well as the immunobooster. We all know it, right? So, but today I'm going to talk about nimbolite, which is neem based chemical, right? So, exclusively I'm going to talk about the nimbolite basically today. So let's actually also talk about the drug discovery and development. Yes, we have been getting the drugs initially from the plants, correct? Talking about the Taxol, which has come from the Taxus brevifolia plant, and Reserpine, and so many other drugs, right? In fact, we got a lot of drugs initially from the plants, and then later we actually moved towards synthetic routes, and even the phytoconstituents. In order to have a larger scale production, we have adopted the synthetic routes. Now this is era of more of a biologics. Every pharma company is trying to work towards the biologics, right? So these are the three sources where you can get the drugs. Doesn't matter what may be the source, but no, you need to prove them scientifically how they are effective. More importantly, how safer they are. And if you don't prove the safety of the compound, never your drug will be going to the next level, right? So, but if you only talk of the isolated compound, but if you talk of the extract, a complete extract, neem extract or ashwagandha extract, we have no problem, you can take it. But as a pharmacologist, if I take extract, I have a problem because one plant can have hundreds of phytoconstituents. I don't know which compound is active and what receptor is going to be. I have to go there. Okay. What compound is going to be active and how it is binding to the receptor, I will not able to tell it. That's why important for me to isolate a compound and do it, right? So based on my observations, I can propose that no, okay, though it is as isolated compound is active, but you can actually thinking of taking a total plant extract if you want to take a long-term benefit from any extract, right? Like a neem. I can actually tell you, if you start taking the neem leaves for a long time, you will be protected from a lot of diseases. And our ancient people, even our older people have been consuming this. And we are been using this, as a toothbrush. Why did we go away from such a, fantastic, benefit? And we know that now we somehow linked to our festivals. during Ugadi, we actually use the neem flowers. So why are we using it? Though it's a tradition, but there is also science behind that, right? But if we can add some more science to that, scientifically, that's what modernization of the uh, uh, our plant system, our herbal system, it's going to give more, the belief to the people, so that you'll be able to convince more consumers, basically, right? Yeah. So see the time, 12 years to 15 years, lot of time, right? Drug discovery is very, very expensive. That's why you have to think of AML now. I think I'm going to talk about a few slides on that, right? So it's a lot of time, but we don't have patience. That's why India is very, very poor in discovering the new drugs. Hardly we have few drugs, and those drugs which are approved are only approved in India. And one drug approved in US takes a lot of time to come to India. Not before 5 years. That means 5 years we are not having a new drug coming to India because of the cost reasons or whatever the reasons it is. So we say that India is a pharmacy capital of the world, right? Even everybody is claiming that. Yes, we have achieved that distinction of the pharmacy capital of the world. If you have so many 7,000 pharmacy colleges, just take one pharmacy college, one objective. How many innovations you should have it? Let's say 100 pharmacy colleges can simultaneously work on one disease area, one drug. Can we come up with up to some level of innovation? If I talk of innovation, it can be a new drug, it can be a new formulation, it can be any herbal supplement which is addressing some of the shortcomings of the herbal natural products. So why are we actually really lacking here? And Sir was mentioning, Dr. Rataya was clearly mentioning it. Research component is really lacking. And I think my faculty colleagues also agree with me. Of course, there are limitations of we don't have enough infrastructure. So that doesn't mean that we will stop working it, right? We need to, from the simple, the thing, tools also you can able to still do it, the wonderful things, right? Small modifications can also bring the new discoveries, right? So it's a $2 billion, lot of cost involved in this. That's why no Indian pharma company wanted to do the discovery. All the multinational pharma companies like Novartis, Pfizer, they all have offices in India, Hyderabad mainly, right? That's why Hyderabad is the pharma hub of the country, and India is the pharmacy capital of the world. So, but what are they doing? Are they doing innovation for the Indian patients? I don't think so. They are doing for the innovation for the Indian patients. They are actually using our expertise just to do the report preparation, data analysis. Of course, we are getting a job opportunities, but is it sufficient for us? Having a 7,000 pharmacy colleges, I was only assuming there's 1,000 pharmacy colleges, but I was told by 7,000 pharmacy colleges, a lot of pharmacy colleges. Just take 50 students from each batch. So number of students we are producing it, number of innovations we are doing, I think there is no match. And if you people don't think why we cannot do and who is going to do this, don't expect your faculty to do it. The thought process should start in B pharmacy. You should have that orientation. Why can't I think of doing the things differently? And it's possible, right? You may have to find the right the problem, or you need to have a right mentor. But no, if you don't want to even think of who is going to come and discuss with you. So students, you need to think differently. Okay, it's to fine to get a job, but more than a job, if you can think of a long time after 5 years or 10 years of a B pharmacy, if you can think of having some idea and having a proper team to execute that, that's where India is going to be leading. Until unless we don't produce innovation like China, you see the China, then all the products, if you see in India, any country, in the US also, every product is made in China. But if you see last 10 to 15 years, probably all the technologies developed by China. If you just use a PubMed or a science directory, these are the scientific engines for you to search the articles. And most of the modern technologies, recent breakthroughs are coming from China. They have realized that just producing the bulk is not going to help us. You need to produce in quality so that you get the patents. Once you have the patents, you can enjoy the royalty for next 20 years. So everything is in your hand, how much you can earn and how to enjoy that part. Just iPhone, right? iPhone, just see the iPhone, one technology, how much their revenue they're generating it. Any technology like that. So that's where I think there is a because we have a huge number of pharmacy graduates going to come up. You can go to US to learn, and you should come back and think up your own discoveries. That's what I want to do it. I wanted to do it, right? Anyways, I'm trying to do my part from Naipur, Hyderabad. So as I already mentioned, these are the different routes to get the drug. And whenever you get a moiety, you have to do the in vitro studies, in vivo studies. I hope that you know what is in vitro, what is in vivo, right? What is in vitro? A cell culture and outside the biology, basically cell culture and enzyme-based assays called in vitro assays. Why you need to do that? And why can't you do the animal study right away? Because number of compounds you can screen are more if you can do in the in vitro. If you have to do the animal study, how many compounds you can do? How many animals you can handle? If you want to study the efficacy of the molecule, how many molecules you can handle at a time? Probably 2-3 molecules. That's why in vitro first, followed by in vivo in the animal models, then followed by you will actually, the data is good, the efficacy is there, if the safety is well proven through your different set of experiments, then only your molecule can go to clinical trials. That's called IND's application, right? These all things will be happening here. So since it's a very risky affair, some companies which are started doing the discovery, they failed at the later stage. If you fail the discovery process at the later stage, it's a big loss to Indian any pharma company, because it's $2 billion US, right? If you have spent already 5 years of time, and you might have already spent probably 1 billion, which is 8,000 crores, is a lot of money for a company. That's why no pharma companies are trying to do it. They are very happy just doing the service again. If you take Sai Life Sciences, if you take GVK, these are all the companies just doing the CRA work, where they take a project from the foreign clients, and they do what are the part to be done, and they get double the profit, and there is no tension for them, right? Worked out or not worked out, enough for them. Just they will say that no, this we have executed the project. This is the outcome, right? India has more than 50 GLP labs. What is GLP? GCP, GLP, GMP, right? So GLP is more important for the new drug discovery. GMP is important for our maintaining the quality of the drugs which are already approved. GCP is during clinical trial, right? Why I'm talking to GLP is, GLP, more than 50 GLP labs are there in India. What they are supposed to do? They have to prove the safety of the drug by doing the animal studies. How many products are coming from Indian companies to do? Why we need 50 GLP labs in India? What are they doing? What kind of business they are doing? Again, we are taking the products from foreign clients and doing it, because very easy for us to do it. Only 1/4 of the cost is required for us to complete the same study, and there is no restriction on the animal usage. Whereas other countries, you have to justify a lot to do, say that no, I am going to use this animal model or this number of animals. So easy for us to do the animal studies, and more easier for the academic settings if we can have a proper collaboration with the industry. That's possible. I was about to ask the PCA president, why don't you make it mandatory to collaborate one pharma company to a college, wherever companies are available in the city or in the state. I think this can be possible, right? Putting a 1 crore money by a pharma company to college is not a lot of money, and that infrastructure really make the college very efficient. And the infrastructure is there. I don't mind. I don't think so. All the faculty not fear of working it. Clearly, the infrastructure is missing in pharmacy colleges. I think that's actually can be proposal. You all can think of it. If not a PCC mandatory, you can actually propose this, right? Pharma company don't mind. They can also get the trained people from the same pharma college, so that they are well trained to work for them further, right? So 10,000 molecules, you will start with target validation, target identification, target validation. So drug discovery is only important for us, right? Okay. I'm not talking about the formulation. I'm going to talk about the formulation, but no, I wanted you to give you some brief. I'm sure you know the drug discovery process, but no, I'm talking about the values. See, you take the 10,000 molecules to 20,000 molecules. NCEs, what is NCE? New chemical entities. 10,000 to 20,000 molecules started. From that, you are expecting one drug to be successful. Who is going to synthesize the 20,000 molecules? Who is going to screen the molecules whether they're active or not? How much time is required for you to do this? And you may not be lucky. Even from 20,000 also, you may not be getting the drug. So that was the problem. That's why it's risky, right? So lead optimization from 20,000 molecules, you probably will be narrowing down to 250 molecules based on the kind of potency it is going to have towards the receptor or the target what you are looking for, as well as the safety you are expecting. There are a range of toxic studies, safety studies you are doing in vitro, right? Which target you want to take it? Which target is good for you? You cannot just take any already existing receptor, right? If like alpha one, beta one receptor, beta receptor, do you want to take the same receptor? Already beaten, right? The receptors, agonist, antagonist already beaten. You need to take a target. Generally, targets are proteins or ion channels. The one which is having a very important role in the disease. The target selection and target validation is very, very important. Right? Like COVID, you know that spike proteins. If you can if you can use a spike protein as your target, so you need to validate that that is only present in the disease and the virus in this case. And it's not expressing in other tissues, right? That's why your target need to be very, very selective towards your disease condition. So target identification is very important, and you need to validate it that no, it is having a role only during disease. And in normal physiological condition, it's not having a role. So you need to have a proper target. So that who is going to give you that knowledge? A pharma company is not going to, get the target right away. Academicians, the student like you, when they perform the experiments and do the lot of omic studies, they'll come up with, okay, this protein, this target is playing a role in this particular disease condition. Now, there is also validation done by some other group. Such kind of targets only pharma companies will take and start working on the, targeting that target to develop the drugs, right? Preclinical testing, 2 to 5 molecules, clinical trials, you all know it. I don't want to really bore on this. And NDA is, once you have a clinical trial data is satisfactory, then only drug will become a NDA, right? Of course, drugs can come back or can be withdrawn from the market even after approval, right? Can you give the example of the drugs which are approved and got withdrawn from the market? Example of the drugs. What is that? We are still using acetaminophen. Paracetamol still we are using it, right? But where is it withdrawn? No, withdrawn means it's never allowed to use it again. Glitazone, rosiglitazone was kind of withdrawn from some of the countries. I think we are using it still in India, but rosiglitazone is not used because of cardiotoxicity. There is a drug called terfenadine, which is an histamine antagonist. This was a classical story. If you can go back and read, terfenadine is a drug withdrawn from the market in 1998 because of the QT wave prolongation, also called Torsade de Pointes. So, means drugs are even withdrawn from the market after approval. How did you miss the safety issues? So what is going to happen now? Since you have missed the safety issues, now you will add more complicated toxic studies to do it. So drug discovery process, that's why it's taking more time. Earlier it used to be 1 billion. Now it is actually going 2 billion. It can even go beyond. Just let me ask one question. If you want to develop the drug for the cancer area, non-cancer area, where is it going to be more costlier? Discovering from starting to end, where it is going to be more costlier? Cancer or non-cancer? This side non-cancer, this side cancer area. Which side it's going to be more costlier for a company? Cancer area, right? Everybody will say because cancer is a costly disease, but no, because cancer area, cancer therapeutics cost you lesser money because risk versus benefit. You don't need to do the healthy volunteers for the clinical trial. You can right away expose the drug to the patients. So you can actually even no need to do some of the toxic studies saying that anti-cancer drugs are already toxic. So a lot of relaxation you'll get in. You can waive up some of the toxic studies. You don't need to do the genotoxic studies. You don't need to do the reproductive toxic studies, not reproductive toxic, causal studies. So many of the toxic studies can be waived up. And no need to do the phase 1 trial. If you have to do the phase 1 trial, right away doing the patients, not in the healthy volunteers. So these are some of the the benefits. That's why if you take many companies are trying to work in the cancer area, because if I have to work in the cardiovascular condition, if I have to work in the atherosclerotic drugs, it needs to be anti-diabetic drugs. You need to take the drugs for a longer time. We don't know the fate of that molecule. 1 year we are taking it. We don't know. It may be slightly causing alteration. And slight alteration, which is irreversible for a long time, can be dangerous, right? So very difficult for to prove the safety of the molecule if the drug has to be taken chronic usage. That's why safe game for some of the companies will be cancer area or short-term medical condition like COVID, right? So these are some of the tools people use it: target selection, lead discovery, medicinal chemistry. Without chemistry, I don't think so drugs can be produced. And again, of course, biology also required. Everybody is required. So it's highly, highly interdisciplinary. You cannot say that, no, okay, I'm poor in chemistry, I don't want to do it. That's how I used to feel in doing B-pharmacy. But today I regret for not focusing on my chemistry. Now I have to purely depending on chemist, whatever he says it, right? So if I would have be having some chemistry knowledge, the way I think my chemistry is different, right? So don't neglect any of the subject. I have seen people, my B-pharmacy teachers, just reading a Renin Martin, you know that? What is Renin Martin? Physical pharmacy book. Just by following that, they have discovered the new formulations. They are actually thousands of crores of business they have done it. Dr. Rambo, Dr. Apte, these are our professors used to just follow the only one book, Renin Martin. And they have produced wonders. Bases are not going to change. By using the basis, you can actually produce the more discoveries, but the basics are not going to change, right? So don't underestimate your basics. Okay? So in vitro studies, in vivo studies, it's okay, same boring. So this is very important. Okay. Kill the compound. So you do all the studies, safety, efficacy, you do it, whether compound results are positive or negative. If you think that the compound or NC is having a problem, killing the compound early is the best approach. If you want to say that no, this molecule is not active, kill it early. Don't let it progress to next 2 to 3 years. What is going to happen? Because you are spending unnecessarily 2 years on the same molecule. Not one person, group of people will be working it. Industry has to pay the salaries and all the things and saying that molecules. That's why, can we predict the safety? Can we predict the performance of the molecule by using modern tools? That's where we have AML can actually be one of the saver for us, right? So everybody's talking about AML. And in drug discovery, it's definitely is going to be playing a vital role. Just go and search in PubMed, AML and drug discovery. You will find thousands of articles. It's not at infant stage now. We have to see the drugs approved. I think recently we have one drug approved, repurposed drug. But no, lot of companies are working on it. So yes, as a pharmacy students, I don't know anything about the AML, but no, don't you want to start this today? You should start it. You should have basics of it. Just having AML knowledge, having your pharmacy basics, B pharmacy basics, enough for you to collaborate with a very rich financial supporter who is going to come to you and say that no, okay, I will fund you. You please do whatever you have to do it. Okay, it cannot happen just in 1 or 2 months of training. You need to have constant efforts. You should start B.Pharm first year or second. Keep on continuing it. AML is going to be very, very vital. So why it is going to be vital is it's going to reduce the time and cost required for the drug discovery. Maybe 2 billion if you can reduce to 1 million or less than 1 billion. It's one good thing. You can definitely reduce the cost from 15 years to maybe 5-6 years. It's going to happen, right? How it will happen is every step. The target you take it is also properly validated through your molecular modeling or protein structures. If you see the alpha folded protein got a Nobel Prize this year. Why they have given a Nobel Prize for the discovery is because once you know the protein structure, you can design the better therapeutics. It can be a synthetic agonist or antagonist. It can be also biologics. It can also vaccine. That means knowing the protein structure is very important. And you know how complex the protein structure is. You have primary structure, secondary structure, tertiary structure, quaternary structure. So many amino acids, there may be modifications. Is it possible humanly to do all the handling? Not possible, right? That's why if we can train the mission through artificial intelligence, it's going to perform accurately. So that's where even target validation to molecular designing, everywhere you can actually have a AML into place. And a lot of companies are already working. What I feel is you can reduce the 20,000 molecules which are required as NCs maybe to 100. That is possible because you are going to optimize everything at the early stage itself. And it's also going to save a lot of time, and probably I will not be failing it because I'm optimizing most other things. Still, you have to prove experimentally. That's a different story, right? Preclinical testing is going to be improved. Lead discovery. What is lead, first of all? Hit and lead. Hit means just going and talking to your receptor is a hit. Lead means what are the properties you want to have is meeting it. Be it a oral bioavailability, be it a stability of it, protein binding, what a PK value, Lipinski rules and all the things they will be putting into rules and say that now how many actually coming positive. And you need 100 requirements. If 90 requirements are there, 10 requirements are not there, it won't go through as a drug because those 10 requirements are problematic for you. How are you going to give the drug? If you have to take a drug every day injection, will you take it? Drug is excellently active. You won't take injection, right? We are already having a problem with insulin. Similar way, so no, you need to maximum, address all the 100 requirements. I'm just giving a scale of 100. It can be more than 100 requirements, right? So this all optimization needs to be done by using your computational tools as well as the AML tools. Advanced version of the computational tool is AML basically, right? Efficient clinical trials. The first the sector pharma industry was using was clinical trials because the amount of the clinical trial data we get it is huge, and it's not possible for us to compile the data. That's why initially they have used in the clinical trials only. They have been using it. Now slowly, in other companies are trying to work on the discovery. Initially it was using the clinical trials. Now it is more of a discovery phases. So these are the some of the AML tools are available. Deep talks will talk about the toxicity because tox is the most important than anything else for a drug to be approved, right? So like that you have a delta vena and many other the tools are there which are actually integrated computational tools with AML. So by using the tools, what are the requirements, be it a chemistry, be it a lead optimization, these all can be done by using the tools, right? And see, every sector US is dominating it. I mentioned, right? US actually take the right technologies and they don't work hard. They actually they are smart. They discover it and have patent it and say that no, oh, you manufacture it. They don't manufacture, right? This is only discover the drug and give you that no, somebody will manufacture and take from you. And at the end, I'm going to enjoy the royalty. That's how their business is, basically. See, the different development phases, many many companies, and these none of the companies are known to us. But these are the AML-based companies which are producing the drugs. These are the compound codes they have given. The target is this, and indication and development phase 2. And this one drug got approved. I just didn't cover here. So possible, right? If we are only talking about the AML, the last 3-4 years, 5 years, how the drugs have gone to phase 2 trial, that means it's clearly indicating that it's reducing the time required for you. And these are the small pharma companies. I don't think so they can actually spend $2 billion. That means they're able to do the discovery in much lesser cost. Making sense? So that's where the AML is actually having a definitely role, and it's going to revolutionize the drug discovery for sure. Collaborations. See the collaborations. All these are major pharma companies, Lilly, Takeda, Janssen, AstraZeneca, we all know, right? These are the dream companies for anybody to work. If you give a job, we all very happy to take it. See, these all pharma companies are trying to collaborate with AI organizations. Why? Because they realized it, okay, whether it's my discovery, whether it's my clinical trial, I need to have a AML, AI actually the expertise. So collaborations are already happening and happening more bigger way now. So they may be collaborating. What is that we can do it? Having a basics. So you just have to learn the basics of it. And all the AML people are not pharmacy people. They're actually purely computer IT based people. But they have no knowledge of the pharmacy. No, if we can also, but AML is not a racket science again, right? You can learn. If we can have the basics of it, the way you think your discovery may be different. That's what, there is opportunity. Me being a pharmacologist for last 20 years or so, 25 years, I joined my master's in 2000. So I'm actually now trying to adopt AML. But I cannot do alone because my expertise is not sufficient. I'm collaborating with somebody else because I don't have time to learn now. But people like you who are just B.Pharmacy completing or masters just joining, you have plenty of time to think of having this expertise with you. Not only for entrepreneurship, not only to do the discovery. If you say that your resume says that no, you have a AML basics. If you can demonstrate to the recruiter, there's no doubt that they won't take you. 100% they'll take you with a very good package. Okay, so think about it. So protein structures are increasing it. Protein structures means targets are increasing it. Means you have more information on the different targets. More information, so more targets are available for me to explore for the discovery. Right? So if you can pull one disease area and one target from existing targets, and if you can understand the target structure and orientation and all, maybe you can also think of doing the discovery at the academic level. Yeah, people are doing it, but I think there is a more rigorous effort is required, right? And just faculty, again, they will have a lot of teaching load. So if we can bring some concept new to them, so it's going to be win-win for both the sides, right? It cannot be just one faculty pushing all the time, right? So you need to have basics, basically. Structure-based design, see, this is the protein structure I'm talking about. So once you have a target, I mentioned 5 lakhs of targets are there. Now you need to virtually screen it. So screening is done where? How do you screen it? On a computer. If you have 1 million molecules, you can screen just on the computer how many are going and hitting it. That's what the virtual screening is. My protein structure, I know it. I'm trying to screen the molecules. Now I'll have few molecules. I'm now going and just having some interaction. That means I had identified the hit. From the hit, now, okay, this chemistry is not sufficient. I need to modify the chemistry while keeping in mind of the requirements of the drug, druggable properties. So you can optimize it. That's called the lead optimization. This all can be done on the computer. By using AML, you can actually more data can be input. If you can give more data, more meaningful output will come, right? That's how the discovery needs to be done. And where target virtual screening through the target protein up to the lead molecule optimization followed by the drug candidate can be coming. So everywhere there is a role. So if you just see the title, earlier it was computer aided, CADD, computer aided drug design. Now it's a computer driven. Driven means every step needs to be computer driven. You cannot just say that no, only designing part I have done it, computer aided. But you need to, if you can apply everywhere, it's going, even clinical trial also, if you can do it, that's what the computer driven drug discovery is more effective. And people are trying to use computer driven discoveries rather just computer aided. That's what this slide is talking about. So this time of 10,000 to 20,000 molecules, we wanted to bring it to 100. Not to start with the 20,000 now. If we can have 100 molecules based on the AML tools, I think you have more time, more chance of getting a better drug. So number of compounds to be synthesized are lesser. Chemistry, only less number of chemistry people are required. Less biology people are required for you to screen the molecules. Since I've already optimized all the requirements, the chance of failing will be lesser. So that's why the total duration of the 2 to 6 years for the discovery and 5 to 10 years can be brought down to less than 5 years, probably 5 to 6 years, because clinical trials needs time definitely. Around 7 years are required. But if by using a proper design of the clinical trial through the AML, you can reduce the time there as well, right? So that is the drug discovery and the role of AML. There is a lot of literature on the internet. You should read it and also discuss with your teachers, right? What happened? Some people are, he's talking some nonsense. We are not understanding what he's talking. So you should ask the questions in between, right? Okay. So as I mentioned, drugs can come from all the 3 sources. I'm taking the natural product is, we are also using a new drug through synthetic routes, but no, one of the favorite molecule is, the plant is nimbolite, which is coming from the neem tree. We know all the properties of the neem. Or Margosa, we have the lemon, we have soaps, right? Margosa company. So that's because of the neem. And every property, people are using this, but these are the list of some of the benefits. People are using it without knowing the scientific validation. We don't know the science behind that, right? But we know that it's effective. Of course, they are effective. But if you can push scientifically, it is going to make more meaning, right? So every book, our Ayurveda, Unani, whatever the Siddha, these all covered in the Charak Samhita and Sushrata Samhita. These are the very ancient medicinal books. They're written in the Sanskrit or whatever language it is. So that knowledge is still useful to us, right? And if you see the government of India, now they're promoting our herbal Indian system of medicine. So definitely one of the plant is going to be neem tree, right? Sarvaroga Nivarani. I feel I have at least done in maybe 6 to 8 disease conditions. I have seen everywhere activity, right? So that means it's the Sarvaroga Nivarani at least up to some extent. 4,000 years, lot of time, right? So this is the neem, and again come from the neem leaves only, right? Very easy. No need to cut the tree, and not like a taxal plant, which is you need to cut the plant. But here you just have to collect the leaves only. Very simple, right? So you can isolate it. These are some of the properties. It contains a lot of chemicals, phytochemicals basically. So one moiety is nimbolite. So we have procured this, we didn't isolate it. Why in herbal is because they are safer, relatively safer, right? That's why we prefer to use herbal. But why are we not taking? Why are we, when we have problem, why are we not taking the herbal? Why are we trying to take the English medicine? Most of the cases, English medicines are then only temporary relief. If you take a paracetamol, it only relieve you pain for 6 hours. Again, it fever will come again, right? So you need to use the body needs to be, body like a doctor, right? If you need to give the phyto constants, which is going to make your body, adapt to that particular stress condition, basically, right? It can be immunity, it can be any other condition, right? So that means if you take a long term any herbal extracts, they are going to be effective. You just expect that no, I'll take today, tomorrow I should have. This is not going to happen in the herbal medicine, basically. If you want that to happen, it needs to be an injection form of the isolated compound. That's what we have done it. Some of the studies. These are some of the conditions we have studied it. One disease area we have done with the ARDS, acute respiratory distress syndrome, also called the SARS, severe acute respiratory syndrome, if it is a COVID virus. So we have an animal model for this, where what is SARS or what is what happened during COVID? We are having people having a severe inflammation in the lung. Lung was fully edematic, fluid accumulation, lot of inflammation in the lung. So that's why people are dying. Right? Because of the inflammation, lung failure was happening it. And if you can manage the condition for 1 week, the inflammation, the virus will automatically go, right? Virus is not going to stay for long time because we are going to develop the immunity against that. But if the severe 1 week of or 1 or 2 weeks of the crucial inflammatory phase is the one causing a damage to the lung followed by other organs. So we have evaluated the protect effect of the nimbolide in the ARDS model, right? This is a pathophysiology where lung will be having a lot of immune cells like inflammatory cells like neutrophils, macrophages accumulating there and releasing the different toxic mediators like cytokines and reactoxin species and causing the damage to the lung. This is a pathophysiology. I don't want to just give the more details. If you see the reactoxin species are there, as well as inflammatory mediators are there, they're actually causing a cascade of events, ultimately causing the cytokines release. Inflammatory cytokines. These are like a virulence in a body. So need to have an animal model, right? So without animal model, I cannot do it. And pharmacologist needs to be little cruel. Otherwise, I will not able to do. So if you see the animal, I need to have a lung inflammation only in the lung. So what we do is we anesthetize the animal. We try to put the contents into the lung. LPS, lipopolysaccharide. What is that? Endotoxin. And endotoxin is an inflammatory mediator. If I put the LPS into the lung, it is going to produce the inflammation. So our model is LPS-driven inflammation, which can also mimic like a COVID inflammation. Right? So, but we have been working on this model before COVID. We have nothing to do with COVID, but we have been working on this lung inflammation before COVID has come. Right? This is the model, and we need to validate that the lung model is properly developed. If the model is not there, having a proper animal model is very, very important. Without that, how will you study the protective act with the molecule? So very, very important requirement is good animal model, right? Very, very essential. So these are some of the parameters. Now, if I can only know the lung condition after isolating the lung, bad idea, right? Because my experiment is all over. I cannot reverse it because I already sacrificed the animal, taken the lung out. Of course, we have to take the lung from the animal, right? So if we can study the lung functional parameters, that's where the technique called whole body plethysmography. Whole body plethysmography is a technique where you just put the lung animal into the chamber, and it is going to give the different parameters, tidal volume, respiratory rate, all the different respiratory parameters are given it. So that's how we study before sacrificing the animals how your compound is protecting. It can be nimbolide, it can be any other agent. So this model, once I have a model, if I have a good chemist, we have designed the molecules and given to me, easy for me to study and just putting in the chamber, induce the model and say that how the parameters are altering it. If the parameters are improving it, that means, okay, my compound is effective. I can able to rank the compound, rank 1, rank 2, rank 3 like that, which are more active, which are less active, right? So this paper actually you can, in the Indian Journal of Pharmacology, I don't want to talk about it. So we also, that's a model. So having a good model is also important. Followed by, we have studied the protective activity of the limbolide in the LPS in vitro. These are cell culture models where raw cells and THP, these are two macrophage type cells. Raw cells and THP cells are the macrophages, which are immune cells, inflammatory cells, which are going to be found to be accumulating in the lung. So we use the cells as in vitro models, stimulated with the LPS and studied how my nimbolide is protecting it. If you see the green fluorescence and red fluorescence indicate that there is increase of the reactive oxygen species. A red color says that it's from the mitochondria. Mitochondria produce a lot of reactive oxygen species. Now, if you give the limbolide, then the intensity is decreasing it. In the protein expression studies also saying that nimbolide is controlling the reactive oxygen species levels, right? So followed by different parameters in the in the lung fluid. So what we do is we take the lung ball fluid, bronchoalveolar lavage fluid. What you do, you take the fluid from the from the lungs and see how many cells are. What we are saying is inflammatory cells are increasing in the lung, right? Number of cells, how much is the protein release, how much LDH levels are increased. These all will be measured. So these are the parameters clearly indicating that normal is some level and the LPS group is also increasing it and the nimbolide treatment was protecting it, right? So once we have this, then we have done histopathology studies in the lung. So normal lung is supposed to be like this, having a spaces for the gas exchange. See the LPS group, there are no proper, the air sacs to have air gas exchange. That's why your lung is failing during inflammatory conditions. So that our approach, our objective is to normalize the lung histology through any medication or whatever the thing. So we have nimbolide, which is kind of controlling the the inflammation model. So different cytokines, which are estimated by the ELISA model, also demonstrated this. We have proven through the Western blot studies. These are not just okay, sorry. These are not just the bands. It takes a lot of time. Just getting one band, band in the sense this is one band, right? Getting one lane takes a lot of time. A lot of optimization required. It's a protein expression studies. So how do you know that okay, this particular pathway is followed based on the expression of the protein studied by the Western blot through the ELISA, through the other RT-PCR, we are going to say that okay, these moieties are altered, right? So we have proven that nimbolide inhibits TNF-alpha mediated NF-kappa B and HTAC nuclear translocation. That means it is targeting the TNF-alpha pathway. TNF-alpha is tumor necrosis factor alpha. It is actually the one of the important mediator of the inflammation. So what is happening? Nimbolide is preventing the inflammation by inhibiting the TNF-alpha. So this is what experimentally we have proven it. Again, this is a fluorescent microscopy where the expression of the NF-kappa B increased in presence of the TNF-alpha, and this was reduced in presence of nimbolide. So computer-wise also, competition also, we have proven that it is actually having a binding. This is the TNF-alpha protein, and nimbolide is actually having interaction with that. So we have proven that it's having an interaction. Followed by, we have also performed the experiments and done few more studies to prove this. And finally, we propose that nimbolide, by inhibiting the TNF-alpha, HTAC inhibition, mediated NF-kappa B translocation, it is preventing the inflammation. So this is what actually the paper we have published in a journal called Cell Death and Disease, which is Nature Publishing Group. It's again freely available. Nimbolide, if you just mention LPS, this paper will come. And very detailed it's given, right? So with a plant moiety, we could be able to publish in such a good paper. So that means we are proving the scientific validation of the compound. If you see the lung inflammation, one of the post-COVID complications is fibrosis. Why people are living? Huh? Okay. Yeah. Can you close the doors? People are going. Okay. Yeah. How much time is there, sir? How much time is there? How much time is? Great. Okay. So yeah, once you have a severe inflammation, constant inflammation in the lung, is going to produce a chronic inflammatory condition and will be causing a normal lung, a flexible lung will become a very irregular, the hard lung, which is called the fibrotic lung. Fibrotic lung is nothing but non-functional tissue, which is not going to be reversible. So inflammation phase is reversible. But chronic inflammation can cause a fibrotic condition, which is non-functional tissue, like a cirrhosis, liver cirrhosis, right? If it's a lung, it's a lung fibrosis, pulmonary fibrosis. So it is one of the complication of the inflammation, constant inflammation. That's why post-COVID, more people are having a pulmonary fibrosis. Okay? This is the link, and we have only 2 drugs approved. And the survival rate, if we see it, only 2 to 3 years. So this is the pathophysiology where excessive stimulation of the inflammation will be releasing certain factors, which is mainly the TGF-beta. This TGF-beta is causing a cascade of events, ultimately causing the deposition of the extracellular matrix, which is nothing but pulmonary fibrosis, right? If you just see the figure, this is a normal healthy lung, where alveolus to the blood vessel, the gas exchange will be taking place. Both directions are important, right? We wanted oxygen to go inside, carbon dioxide to come out. So if you have this is the barrier, but see the barrier is increased because extracellular matrix accumulation. That means more extracellular matrix accumulation. So pre-excision of the gas is actually not happening it. So that's why we have a problem with the lung, right? So we have studied how the nimboloid is effective, because we mentioned that ARDS, short-term inflammatory model, it's effective. Even in the chronic inflammatory model also, nimboloid was found to be effective. So these are all the different experiments, cell culture-based experiments, Western blot experiments, as well as the tissue experiments. Alpha SMA is one of the marker for the extracellular matrix, found to be increased expression. This brown color is increased in the bleomycin. What is bleomycin? It's an anti-cancer drug. Bleomycin is an anti-cancer drug, but side effect is pulmonary fibrosis. So we have used anti-cancer drug to induce the model here, right? So like that. So even for you to another model, we can use another chemical. So inducing agent should be having a particular pathology to cause the change in the tissue. So whenever you do the study, what you do, you will have a normal condition, disease condition, followed by treatment. What I expect, my disease condition will have abnormality in the tissue or the cell. And when I give the treatment, I expect the tissue should be having improvement, if at all, if you want to claim that drug is active. That's the only, any pharmacologist, any pharmaceutical research objective is this only, right? You wanted to protect from the particular condition. It can be a cancer, it can be fibrosis, it can be any other condition, right? It can be hypertension also, right? So autophagy was one of the concepts. There was a Nobel Prize again given to it. Autophagy is a kind of cell death, where not like a necrosis or apoptosis. Autophagy, some of the organelles which are malfunctional will be eaten, self-eaten basically. So this autophagy is a process where it was found to be abnormal in the pulmonary fibrosis. Nimboloid was causing a autophagic based clearance of the abnormal structures. That's why it is protecting from the pulmonary fibrosis. That's what we have proven it here. Again, this paper we can just find it in this publication, right? So lung is over, lung fibrosis is over. Just see the title: nimboloid ameliorates unilateral urethral obstruction induced renal fibrosis by inhibition of the TGF-beta, and the title itself is talking about what it's saying, that it is protecting against the renal fibrosis. We know the chronic kidney failure is one of the biggest problem in India, throughout the world also. And maybe if we can take the neem leaves, we never know, because chronic kidney failure is like a silent killer. We'll only come to know the kidney failure after 50% of the kidney is gone. Hypertension is one of the contributors for this. Diabetes is one of the contributors for this. Why can't we take our medicines, which are plants? Just take it. Whatever the way you can take it, and you at least preventing the incidents of the kidney failure, right? Any condition, basically, right? So for you to believe that, you need to do the experiments, right? So that's what we have done. Again, this is a detailed work, but I just only kept the abstract of it, because of just to say that it is even effective in the chronic kidney failure condition, right? Renal fibrosis. How do you induce this model? Again, ureter, we have abstracted it. Once you abstract it, the urine flow is obstructed. That's why it is causing a pressure, stress. Somebody was talking the stress can also cause a cancer. Stress can also cause a lot of abnormality in the tissue. Exactly happened in the renal tissue. And it's well known UUO model. UUO model is the renal fibrosis model. And this model is surgical model. Anybody can develop this simple model, right? Another model, see what nimbolite abrogates cerulean induced chronic pancreatitis. You know, acute pancreatitis, more than 50% mortalities. Alcohol, chronic alcohol induced can cause acute pancreatitis. Once acute pancreatitis is there, 50% mortality. Just see the history of the chronic acute pancreatitis. And chronic pancreatitis is another complication of the chronic irreversible abnormality in the pancreas. Pancreatitis is small tissue. Steve Jobs was diagnosed with pancreatic cancer, if I'm right. So chronic pancreatitis became a chronic cancer later. So link it, right? So even it was protective. See, this is the mechanism we have proposed it. Whenever you say that no compound is active, just doing one study, one parameter is not sufficient. You have to explain molecularly with the proper pathway that no, these are the markers altered, these are the markers increase or decrease, then only your paper will be accepted. Without doing these studies, nobody will say that no, nobody is going to believe it. No paper will be accepted. And this is actually almost a 10 impact factor paper. Very difficult to publish it, but no, we could able to convince it by proving all these pathways. What I'm trying to say is, you can actually enjoy doing a lot of molecular studies, then publish it, right? This in cancer. Curcumin, we say that anti-cancer. But curcumin, you need a 20 micromolar dose. 20 units. But nimbolide, you only need 2. Which one will take it? Will take 20 mg or will take 2 mg? 2 mg. So I will say that no, much better than curcumin, nimbolide. Much better than, 10 times better than the curcumin. But how many of you believe that nimbolide is anti-cancer? When we published this work, I didn't even know that no, Times of India has covered this work. The Times of India has covered this work. Not for this work. Along with this work, I have also proven that no, nimbolide, if you want to use as anti-cancer, to treat cancer, you need to take injection because the oral viability is an issue. So we have, I just presented this. So, but if you do a good work, you don't need to advertise it. People, there are people to, if they have cancer patients come to my lab. Literally, the relatives came, they wanted nimbolide. They want to use it because they are in the certain stage. But as a pharmacologist, only doing animal studies, do I have the right to give the molecule? If I just tell them to take it, if I give the moiety, the nimbolide, what are the consequences, right? So I'll be completely, I will be, punished. So I have to give the proper, see, I cannot give this because there are a long way to go from there. But as an experimental people, at least we need to prove that it's effective. Then somebody will take this idea, right? Yeah, it was effective basically. What again, this paper is published. It was effective in the cancer as well, much better than some of the standard anti-cancer drugs. If you take a cyclophosphamide, its IC50 value is 10 to 20 micromolar. Cisplatin, its IC50 value is highly more than 20 micromolar. But cisplatin is toxic, nephrotoxic. We are still using the drugs. That's why people say that no, it's not the cancer which is killing the patient, it's a cancer drug which is killing the patient. I 100% believe this. Take any cancer drugs, conventional anti-cancer drugs we are talking, Taxol, you take the doxorubicin, you take the cisplatin, these are all highly, highly toxic. Not just one organ, all the organs are getting affected. So that's why you don't have proper the medication, at least in India. CAR T-cell therapy is very good. But how many of you can afford this? Is it technology available in India? Immunotherapy people are talking about. How much immunotherapy moieties are available in the country? We don't have. Most of the people are only again taking the same drug, which is conventional anti-cancer drug, which are highly, highly toxic. So what I'm trying to convey is there are medicines which may be further developed. I cannot develop alone. My job is just to prove as a pharmacologist to prove that it's active. But this needs to be further taken up, right? So this is right. It's osteoarthritis basically. We have even proven it because if you see the TNF-alpha inhibition, anti-TNF-alpha antibodies are given in the arthritis. If you see the arthritis, anti-TNF-alpha monoclonal antibodies are given. But monoclonal antibodies are very, very costly, right? The one injection probably cost you 20,000 to 50,000 rupees, one injection. And but may not be able to afford it, right? But the same TNF-alpha can be inhibited by nimbolide. So we have studied this in the arthritis models, osteoarthritis. This is a normal joint, and if you have arthritis, the tissue will be abnormal and all the erosions will be happening. We have done the animal study, how the nimbolide is effective, and see how the rat is walking, and all the studies we have done it, how the arthritis scores are altered, what they are all indicating, and how the walking pattern. The animal is not able to properly walk because it's a joint inflammation, right? We have proven that nimbolide was effective in the arthritic model. Even if you do the X-ray also, you have to do it because to see what kind of joint and bone changes are happening. These are the X-ray images of the thing, and see cartilage which is intact in the normal is lost here, right? There is no cartilage, and treatment with nimbolide, we can see some kind of improvement is there. And these are the papers. First paper is talking just about the alone, but see the second co-treatment of the nimbolide augmented the anti-arthritic effects of methotrexate. What is methotrexate? Again, we are using this as an anti-arthritic therapy, immunosuppressant. But methotrexate is a very toxic drug, right? So I'm saying if you combine it, it's going to improve the activity and also reduce the toxicities. Is it not a good one? That's how it is. But that doesn't mean that you will take a neem extract and methotrexate. There is going to be a lot of drug-drug interactions. You need to have a proper regimen done, right? Right? Psoriasis is another model. So all the immune-driven models that's why it's a server nivarani, right? So we have done the, so this is psoriasis where normal epidermis, you see the epidermis thickness is increased. And there is a kind of decrease in the epidermal thickness. That's the psoriasis, right? And again, we have a model for this. Imiquimod is the inducing agent, basically. We have seen this. And again, this is the work published. But story is not end here, right? What is happening is nimbolide is excellent. There are 5-6 papers. I only did it, right? I have got enough papers. Okay, I'm happy. But no, nimbolide has a problem. See, this is not orally bioavailable. And I don't want, nobody will take the injections of nimbolide, right? Extract, nobody will inject it. Even nimbolide also, will you take the drug alone? But injection is not very feasible one, right? That's why it has a problems. There is a need to develop this. So, see, poor oral availability, pharmacology activities are seen only with the parental administration. This is one of the drawback of it. That's where a medicinal chemistry will jump into this picture. The formulation comparison needs to jump into the picture, as well as the computational biology can also jump into the picture. Why computational biology is? Computational biologists can say that, okay, these are the ways you can improve the oral availability. You can improve the solubility. That's where they can work. So, lot of publications. I only did lot of publications, right? So, with many natural products, lot of publications are there. However, the problem remains same. We are not translating to the next level. The translation needs to happen, can only happen if we have all the teams coming together, believing that, no, what are the pharmacology I have done is making sense, and next person needs to take the battle and take it. Otherwise, it's going to happen one group doing it, over, right? Just see the pharmacokinetics. Concentration in the y-axis, nanogram per ml, only 25 microgram, 25 nanogram, whereas IV is 5,000 nanogram. That means if you give nimbolide intravenously, just to study the pharmacokinetics, very good, 5,000 nanogram concentration is there. Whereas oral, only 30 nanogram maximum. That what it's saying that no, not the, and how much dose we are given? 50 mg per kg, a lot of drug orally. That means it is having a oral availability issue. That's what we have proven it. These are pharmacokinetic parameters. Just see the percentage oral availability is less than 2%, 3%. Even this is higher side, I say. So that means if you take 100 mg, only 3 mg is going. That is not sufficient for you to prove it. Where if you want to treat the condition, medical condition, that's where you can use them as extracts. For long term, before the disease has set in your body, if you take it, only less concentration is required for you to get a benefit. Once you have a disease already established, like a cancer or any condition, there is the only way of is you have to make a new formulation and take injections. Just take, otherwise orally is not going to reach the sufficient concentration. That's what we have proven it here. So these are some of the approaches we have done. We have done the formulations. We have made the liposomal formulation of the nimbolide and demonstrated that no, they actually go to the lung sites where inflammation is there. These are some of the experiments we have proven it. And this is a paper we try to publish during COVID. And we say that no, this is when we even use the COVID associated, but they didn't agree on this. But we tried a lot, but finally during the COVID only we have published this work saying that no, IRGD conjugated liposomes, like we made a liposomes, are actually effective because we know that no lung, we need to protect from the lung, and tocilizumab is one of the drug we have been using it because we are believing that no, that drug is going to be controlling the inflammation, right? So, but no, we are actually saying that no, this nimbolic can also be effective. But no, ultimately we have published in another journal. This is the histological data we have provided. It's not moving. Can you go to next slide? I think I have only few slides. Can you just quickly go to the next slide? Next slide, please. Is it stuck? Huh? How much is time? What is the time now? Huh? Okay, I'm done. Just few more slides. Can you go to the slide? Okay, so yeah. So this is one formulation we have done it, right? Another formulation. Okay, no. Yeah, I'm not done. I'm not done. I'm not going to leave you people. I'm not done. 5 more minutes. Not for me, for you only. Okay, yeah. What is that? Okay. What happened? Okay, so before actually, till we get this, what I'm saying is, okay, we demonstrated arthritis, right? But again, we have given it intraperitoneally. That means IV intraperitoneally we have given it. So what we made was, we made a nimboral microparticles, PLJ microparticles. We know microparticles release the drug for months together, at least 3 to 4 weeks, even beyond it, they will release. What we made was, we made a nimbolide loaded microparticles, we injected into the joints. You know, some injection is taken during the arthritis. What is the injection we take? Hyaluronic acid gel we are taking. When you are taking the just gel, which is only for the cushioning effect, if my nimbolide is effective to control the inflammation, why I should not take the nimbolide injection of microparticle? One injection is sufficient for me to control 1 month. That's what the approach we have tried. Single injection of the nimbolide microparticles are reducing the inflammation for 1 month. So when we compared that with the free drug nimbolide given every day. So that is another approach we have done it. Yeah, so since it was the under psoriasis, we made the niazomes, we published this work. Again, I'm skipping this. Microparticles I'm talking about, right? See, the drug is releasing up to beyond 30 days. That means slowly, drug will be released. I'm putting as a depot in the joints, and it's going to release the drug slowly. That's where we say that no, microparticle based the application into the joints can be effective. So we have proven that experimentally. Again, in the arthritis model. These are the histological studies, again to prove it. Again, this is the paper we have published. You see the title? I think you are not able to see this, right? Okay. Anyways, so these are the nimbolide loaded sustained release microparticles as single dose formulations for effective management of arthritis. That's why single dose is sufficient. Just you have to just take like a hyaluronic acid injection. Maybe you combine hyaluronic acid along with the nimbolide, you take it. But again, we cannot right away take the patients. We need to do the required studies to take further, right? Whatever it may be, whether it's a formulation or the natural product or the synthetic molecules. See, safety is more important. Toxicology studies are highly essential. See, safety is important than the therapeutic activity of the drugs. These are the toxic studies to be done. That's why you need to use approximately 20,000 animals for the new drug to be successful. But out of 20,000 animals, 18 to 20,000 animals just go for the toxic studies. You only need 1,000 animals for the pharmacology studies. That much important is toxicology studies. Many of the drugs fail because of toxicity. That's why having a knowledge, ICSQSEM, S stands for safety guidelines. If those we want to understand the safety aspects of the drug, which is the safety guidelines, ICS safety guidelines, you can just go through it. These are the guidelines, clearly mentioned what dose to be taken, how long you need to expose. These all are given in this particular the one. As I mentioned, you can actually predict the toxicity without performing the studies. That's why you have a in silico toxicology studies. This is one area where there are tools to say that whether the molecule is probably toxic or not. You can predict it by using a just computer based design can be. This is a summary. What we have so far seen is given here: nimbolide, which is anti-inflammatory because versatile pharmacology properties is experimentally proven. But no, however, We have to go, see the clinical trial or whatever it is required because we need a larger quantities. And there is a need for us to produce a bulk quantities of nimbolide. And nobody, actually, it's very costly if we have to do the experiments. But there has to be approaches to produce larger quantities where pharmacogenesis people can think of producing a bulk quantities. And when I asked the company, why are you producing nimbolide? What purpose? They're actually sending this to Germany. And we know that we are saying that no, nimbolide is, curcumin patent rights have gone to US. Tomorrow, nimbolide patent rights is going to go to somebody else if you are not protecting it, right? So we need to be aware of this. So with this, I would like to thank the organizers and thank you for your patient listening. Thank you. You have any questions quickly? I also have a train to catch. Any questions? I'm glad at least I could hold all the people. Thank you. So any questions quickly while the formalities are happening? No questions? Thank you.
So any question? This session is now open for discussion. So I would like to thank sir for his insightful presentation. So I have a query, sir. So what made you to pick up nimbolite out of complex molecular diversity of neem?
So if you see the literature of the most of the famous national Indian plants, ashwagandha is one of the natural plant and natural product which is highly explored for the pharmacologically, and the neem also one of the widely explored natural product. And the major constant of the neem is nimbolide. Whereas in the ashwagandha, which is Veda ferniae, as a pharmacologist, I just want to pick the which is the most active moiety. That's why I have taken nimbolide. This is the most active for the pharmacological properties. If we talk about agricultural applications, azadirachtin is the more important for the neem tree. So that's why I have taken nimbolide as my moiety. I would have taken another moiety, but this is most prominent pharmacologically active moiety reported already in the literature for the cancer. So based on that, we have taken that moieties. Yeah. Thank you.
Okay. Thank you. Thank you so much, sir, for your valuable expertise and thoughts sharing with us. Thank you, sir, once again.
I now request Dr. Jitendra sir, Dr. Midun Rudrapal sir, and faculty delegates to felicitate Dr. Chandra Godgo sir. I request all the faculty delegates to please come on to the dais. I now request the delegates to felicitate Dr. Midun Rudrapal sir. Sir, please. A small announcement. Someone has lost their key in the Vignan Pharmacy College premises. Owner of the key, please come and meet me. The cultural session will start after 5 minutes break. Snacks are available outside the seminar hall. Please have them.
అక్కడ ఎవరున్నారు అడుగుతుంది అన్ని రావు మళ్ళీ నా పేరు వస్తుందా సరే ఇది కాలేజ్ సెట్ చేసుకొని సీనియర్ వెంకట ఉంటుంది వెంకట కూర్చోడానికి కలిసి సెట్ చేసుకుంటాం మనకి అవసరం లేదు ఇదే మీ ఆడియో
మధ్యలో చాలా చేంజెస్ చేశారు స్టార్టింగ్ లో అన్నిటికీ
ఇదే
ఇప్పుడు
మహేష్ గారు పెట్టాడు మహేష్ పెట్టాడు మా ఇదే కదా పాట స్టార్టింగ్ పెట్టా కదా ఎన్ని మినిట్స్ అది చెప్పు పక్కన పోయి రాపో త్వరగా ఇది కాదు సరిగా ఒక 5 నిమిషాలు టైం రన్ అయ్యే టైం పెట్టి డౌన్లోడ్ చేస్తారు అది కూడా చూసుకోండి ఎందుకంటే ప్లగ్ అరే 8 అన్న 8 పాటలు 16 minutes 21 minutes రేపు ఇంకో రేపు రేపే రా ఫిల్మ్ కల్చర్ మేడం ఒకసారి వినేలు ఒకసారి వినేలు కదా కరెక్ట్ చేసింది నేను కాదు అదేనా రావట్లేదా సరే అదే పాట అదే 16 దగ్గర నేను దా పెట్టిందేగా ఫ్రెష్ గా అదే
సరిగ్గా బాగా వేయండి
అరే వాడు ఒక్కడు పిలుస్తున్నాడు వెంకట నువ్వు పెట్టినాడు వెంకట అని పెట్టావే వెళ్ళనే వాడిని పిలువు కాస్త
పర్ఫార్మెన్స్
8 ఉన్నాయంట ఫ్యాషన్ షో 6:30 7 ఉన్నారు కదా సరదాగా వేయాలి కదా
ముగ్గులేదు ఇంకా ఫస్ట్
మరి 3 రోజులు 3 రకాలు అన్నారు
ఫస్ట్ డే
తింటావు సార్ ఒకటి ఉంది ఏదో చేసి పనులు వస్తాం
ఆకలి వేసినప్పుడు ఒకటి తినేయండి అక్క
ఒకసారి మర్చిపోయినా నీ గురించి ఫోర్త్ ఇయర్ అయిపోయింది
నెక్స్ట్ ఫస్ట్ ఇయర్ బాయ్స్ ఫస్ట్ ఇయర్ గర్ల్స్
ఫస్ట్ ఇయర్ బాయ్స్
మధ్యాహ్నం ఫుడ్ కానీ పకోడీ నచ్చాయి
చికెన్ తిన్నావా
బాగా నీళ్ళు
పన్నీర్ బాగుంది అయితే తెలిసి
రేపు కూడా చూసుకోండి అయిపోయినా సర్ది చేయిస్తారో లేదో. అందులో తీసుకోండి అవును మీరు ఈ డ్రెస్ చేశారంటే మీరు ఎంతగా వస్తారు అనుకున్నా ఎవరు మీరు చెప్పాలి కదా ఇన్విటేషన్ ఇవ్వలేదు ఇన్విటేషన్ పెళ్ళికి ఇన్విటేషన్ ఇవ్వాలి కదా ప్రింట్ ఇచ్చారా
లేయరింగ్ అని చెప్తుంటాను అనమాట నా లేయర్ మహేష్ అండ్ జాయ్ సిటీ మన మెలాంటి పైకి వెంకటేష్ గారు ఆర్డర్ ఎవరు ఏ పర్ఫార్మెన్స్
క్లీన్ స్వీప్ చేశారు
ఏంటి అరే అది లైవ్ వస్తుంది నీ మొహం నీ మొహం వస్తుంది మళ్ళీ యూట్యూబ్ లో లైవ్ వస్తుంది
మరి వాయిస్ ఆడియో ఆఫ్ చేయలేదే
అక్కడ మైక్ లో ఉంది అక్కడ కనెక్ట్ చేసింది ఒక నిమిషం వాళ్ళ పేరే పెడుతుంది మర్చిపోయాను శ్రీలక్ష్మి ఫైనల్ ఇయర్ మాది అయిపోయింది
నెక్స్ట్ ఫస్ట్ ఇయర్
వాయిస్ గోల్స్ అయిపోయిన
తర్వాత ఫ్యాషన్ షో స్టేట్
ఒక నిమిషం ఫ్యాషన్ షో ఫ్యాషన్ షో శ్రీగారు పిలిచాడు గోల్దంగా రా అని ఎక్కడ రా తిట్లేదా ఈ రోజు కాదుగా ఆవుపోతే
అందరికి అదే చెప్పు
ఫ్యాషన్ షో తర్వాత
థర్డ్ ఇయర్ సుజుకి వాళ్ళ టీం
ఈ ఆడియో ఫైల్ పెట్టాడు
ఫ్యాకల్టీ 2000 అన్నట్టు అనిపిస్తుంది నాకు వెనక్కి రా. వస్తా వికిల్ లేడు వికిల్ ఎత్తుకుంటే ముందుకు వచ్చా వికిల్ గడ్ బయట ఉన్నాడు ఇది కనబడటానికి మాట్లాడుతున్నా
అసలు లేదు
ఇది
అది శ్రీ విద్యది
అయిపోయిందా అది
ఛార్జింగ్ ఎందుకు రే వాడు మనకి చూపించడానికి తీసాడు ఇప్పుడు ఛార్జింగ్ చూపించడానికి అదంతా అక్కర్లా
ఎవడు ఎవడు చూసాడు రా
నాకు వాడు ఫాస్ట్ తెలుసు నీకు తెలుసా అది చూపించాలి
రాజు చూస్తున్నాను
ప్రిన్సిపల్ ఫోన్ పాస్వర్డ్ తెలుసు
అదే ప్రిన్సిపల్ ఫోన్ డీటెయిల్స్ ఉన్నాయి కదా
తీసుకెళ్ళి కొత్త మధ్యలో పెట్టి ఓటీపీ రాకుండా దాన్ని ఏం చేయలేము మా ఫోన్ తీసుకొచ్చినప్పుడు చెప్పు మనం ఏదో పొందాం
మాస్టర్ కార్డ్ కూడా కావాలి
అది
అప్డేట్ చేయడం నా మాస్టర్ కార్డ్ చేసా చాలా కింద రూపాయికి వెళ్ళాం సెకండ్ అదే అవరా ప్రిన్సిపల్ వేసా కడికి అదే అవరా
వీసా క్రెడిట్ కార్డా డెబిట్ కార్డా
నేను వేసా చూసి అంత డీటెయిల్స్ చూడలేదు కార్డు
ఇందుకు వస్తుందా
థర్డ్ ఇయర్ తర్వాత శ్రీలక్ష్మి
గర్ల్స్ బాయ్స్ అయిపోయింది ఇప్పుడు గర్ల్స్ చరిత గారు రోషిని గాని పెట్టుంటారు
పెట్టలేదు నాకేం పెట్టలా నువ్వు టెలిగ్రామ్ పెట్టింది అది ఎవరిది
అది గ్రూప్ సింగింగ్
గ్రూప్
సింగింగ్
సింగింగ్
అది ఎక్కడ అబ్బాయి దగ్గర ఉంది అబ్బాయికి షేర్ కి అందులో వచ్చింది అంటారు దేంట్లో
ఫోన్ చేసి పెట్టి
ఫ్రీడమ్ ఆల్ట్ వచ్చేసారు లైట్ గానే ఉంటుంది సేవ్ చేసి పెట్టుకున్నావు
అరే అది స్ప్లిట్ ట్రై చేస్తున్నావు రావట్లేదు దీనికి ఎట్లా ఇచ్చారు ఇదిగో ఎట్లా ఇచ్చారు ఆన్ చేస్తుంది ఇది వస్తుందా నాకు మాల్ వస్తుంది ఫోన్ చేయ నాకు అక్కడ కనపడుతుంది బ్లూలోనే వస్తుంది అది ఏం చేయాలి నాకు అర్థం కావట్లేదు ఎక్స్ట్రా ఆప్షన్ ఉందని అంటే బ్లూ కలర్ లోనే ఉంది అది తెలుస్తుంది అంటే చూసుకో పన్నెండు రాదు సరే సరే రోషినికా ఫోన్ చేస్తారా ఫోన్ చేస్తావా బొకే బాగుంది బొకే తీసుకెళ్ళి ఆడియో ఆడియో అంటే ఆపే అన్నా ఆడియో కూడా రికార్డ్ అవుతుందంట ఆపేయండి ఆపే అన్నా మైక్ ఆపే మైక్ ఆపే